Hi everyone, welcome to the sixth dive of our Climate Connections at the IC Interface expedition. Today we are diving off the coast of the Bach ice shelf of Antarctica. We're on the eastern side of the Ronne Entrance. Our plan is to dive down to around 950 meters depth and work our way up a slope to around 650 meters depth. It'll be a longer dive. Today we're going to recover at around 5 a.m. local time. And we just started the dive here a little bit ago, so we're looking at like a 13.5-hour dive or so. My name is Erin Easton. I'm an assistant professor at the University of Texas, Rio Grande Valley. And I'm also here representing Ocean Census, and this is a Challenger 150 supported expedition. When we get to the bottom, I think we're going to be focusing on taking some push cores if it is sedimented at the seafloor. And then we'll work our way up doing some transecting and faunal collections. So hopefully we'll see some really interesting fauna and a diversity of fauna. That's our plan for today. So we still have a few hundred meters here until 250 or so meters until we hit the bottom. So I think we'll probably go back on mute here, and when we're on bottom, you'll probably have someone else in lead. Hi everyone, so we're back off mute as we continue our descent. We have around 100 meters to go. Yeah, we're doing science party shift change, but we just got a new schedule, so I'll be with you for a little bit longer, I think, until the next watch lead can come in. So right now, in the room we have Soph on streaming control.
Hello.
Zach and Tyler are flying and Ominous. Tyler is flying. And we've got Pedro, Benjamin, and Gina in the room as well for the science party. And yeah, on the way down we've, let's see, saw Siphonophore, I think was streaming for you all. Tinaphore. Yeah, we saw Shrimp.
Oh, yeah.
And unfortunately, I don't think we got the stream up quite in time to catch the shrimp, did we?
No.
No, sorry about that. Maybe we'll see another one. Tyler loves shrimp, so very photogenic.
The first part of the dive is called shrimp hunt.
That's it. Be no more shrimps.
We're just, we're just talking about lunch because, you know, we're obsessed about food on this
Jellyfish,
ship.
catfish.
An amazing lunch. Avocados,
Reach out and goose this
spinach
fish.
blue cheese soup. We're chasing down a fish. See if Tyler can catch it.
Go, Tyler, go.
There we go.
So if Tommy's not here, the other can just take it.
Okay, that's fine. Yeah. Okay. Yeah, so yeah, I think Pedro is gonna take over for me here while they're once we get on the seafloor. Oh, one more shrimp.
Oh, I hope you don't go too far. Erin, can you tell us a little bit about why we picked this specific location to dive?
This location that we're at, we did the first mapping in the area, so no one's ever mapped here before. It's an area that's been not studied then either, so it was of interest to us to come over to a mapping. Once we did the mapping, the plan is to go upslope from one of the deeper points up to one of the shallower points. So they found an area in the mapping that was of interest geologically for our geologists on board, as well as has some nice features on it that we think will be interesting for the biologists in terms of the faunal perspective. Hoping to see a diversity of fauna. So that's why we're diving here. And this station that we're at is station 8, so we're at part B of it. We were originally just going to do one station here and kind of map in one particular area, but we decided it was really interesting when they started doing the mapping, and so they divided it into 2 stations. So it's essentially 2 stations that we're doing our complete set of sampling at. So we completed station 8A already, and now we're at station 8B. Yeah, that's the general plan.
Amazing.
It looks like we're about to hit the sea floor, so I guess I'll let Pedro take over here, and he can introduce himself and the next rotations team. Thanks.
Perfect. Thanks, Erin.
Yeah, no worries.
Okay.
Oh, we're still not getting altimeter. Going down into crevasse.
Which steps we expect here?
The dive plan said 9:58.
Ah,
Oh, yeah.
Almost
That'll be
there.
about that, yeah. Now we're getting
Yeah.
camera and show the icebergs a little bit, if there are any. Oh my goodness, so salty. Ah,
Some glare, huh?
that is quite some glare.
Wow. Camera doesn't look like it's very happy.
No.
Oh,
Go, go.
that's the bottom.
Okay.
All right.
So we see bottom at 945 meters. And the first thing we see is a holothurian. This is sea peak or sea cucumber. We see also a number of holes in the sediment. This is produced by worms. These are tubes from worms. There is some fish, and I think that there is also, is that an octopus in front of us? Maybe a small one, and a fish. So it looked like a very nice place. It's deep. It's one of the deepest area that we have found here. And we see that it's relatively flat. Ah, okay, this is one of these fishes. What we can do is just to transit a little bit, see how it looks like, and then we will stop for the bush chorus.
Not that.
That fish is of interest of one of our colleagues.
That's true. We could slurp this fish, you know, with the slurp gun. I mean, not that specifically. I don't know if we will find it now. In case it's there.
Means slurp it or any other.
By any means necessary.
In case we don't find this specific one, we can move.
Trying to find. Is that it down there, you think?
So we have produced a little bit of disturbance. The sediments here at the bottom, they are very fine. And of course, every little movement of the water is producing that we are generating like a sediment cloud. We have seen at the beginning a small fish. This is one of the fishes that we are very interested in collecting here from Antarctica. And we are looking now if we find it again in order to stop and try to collect it. That's probably the sea pig that we have seen at the beginning. And we will see. There is in the water, there is a haddock nut swimming. We are moving now slowly so that we don't disturb the bottom. This looks like a typical deep sea sediment. Deep sea bottom. Again, we are almost 1,000 meters depth, and we see a lot of organisms here actually. There is also some crinoids. There are a number of sea cucumbers. These roundish red structures. This seems like sea urchins. There's an ice fish that we're going to look now. It's a lot of life here. I didn't expect that. I thought that it would be less life. But these depressions, these little areas which are deeper than the surrounding seafloor, they accumulate. They tend to be kind of a trap for sediments and for algae that are coming from the water column from the top. And this means that there is actually a lot of food for the benthic organisms at the bottom, for the organisms that are living at the bottom. Okay, can we get some lasers?
Yeah.
So that we can, with the lasers, we can estimate the size of the organism. So they are, if I'm well informed, 10 centimeters apart. And then we can estimate how large the animals are that we see. We see some jellyfish.
Continue on transit.
Okay, yeah, continuing transit. We want to just get an impression how it's look like here. What we can say at the moment is that we will be able to collect some holothurian, some sea cucumbers. There's also anemones, like the one that we can see here at the left side. We see also some corals. And what we want to collect here at the deep station is also sediments. The sediments are used to characterize the environment, so we want to know the grain size, but also how much food is in the sediment, so the chloroplastic pigments, the total organic carbon in the sediment. We look for small organisms, for foraminifera, that are used to reconstruct the temperature of the waters in the past, but also we look at the small organisms that are living in the sediments, like small worms. We have also crustaceans, like little crabs. We have different kinds of organisms inside. We see now swimming in the water, there is a worm, it's a polychaete. They use the water currents to be transported by the currents, and so they can colonize different places. Well, this is nice. I think that we got an impression of how it looks like here, and we could stop in order to get the push corals. There is now like a polychaete there, or I cannot see if a polychaete or a fish.
I think that's a fish.
I think it's a fish. Huh?
Alright, we'll stop here.
Yep. So we are maneuvering now to make a stop. And the first thing that we are going to do is to collect the sediments with the push corals, and then we will have plenty of time to look for different kinds of animals, to collect them. Well, sorry, I didn't introduce myself. I am Pedro Martinez. I am working in Germany at the Senckenberg Institute, and for many, many years, almost 30 years, I'm a marine biologist. I'm very much interested in deep sea, and also I have been to Antarctica many years ago. This is my second time. Antarctica is always amazing because of the amount of life that is here. And it's a very remote place, so most of the organisms that we find, at least the smaller ones, they are new to science. So this is for us very interesting and very important to study.
transect. You want to push all the way out?
What are we doing? We're doing.
I think approximately 955 push forward.
Put it out. Put it out.
We doing all 20? Here.
Yeah. No, excuse me, from Fortaleza. We can start with 18.
18?
Yes.
Okay. And different groups want different amounts, right? So biologists just want the top.
The first 5, we want to leave a little bit of water.
Okay.
I will tell you.
Okay.
Yeah, first 5.
Just tell us whatever you need.
The first 12 really is a couple of centimetres of water at the top.
Yeah.
And they're the standard sized ones.
And the long one goes all the way.
Yeah, the long ones as full as possible.
Okay.
I'd rather go second, but I can go with everything.
Bridge, ROV.
Go ahead, RV.
Hey, we're about to do some push course. Is there any threat of ice making us move anytime soon?
Yeah, we've got one off the port side. I am tracking it, but I might have to move a little bit more to the north.
Roger that. Thanks, Jason.
I'll get back to you in a second.
You got it.
We've got lots of options if we want to preempt it.
Amazing. Sorry, Tom, I didn't see you there. Good morning.
Good morning.
Wouldn't take how much it is.
Morning all.
Is that making that one? Yeah.
I think so.
Yeah. Okay. Yeah, we had a little problem with the mic, with the microphone.
Right. You're not drifting dirt all
Hi
over.
all, I'm Tom Linley. I'm a fishy guy from the Museum of New Zealand, Papua Tongarewa, and I've just woken up, so bear with me.
RV bridge, probably be a bit more comfortable if we could move sort of 50 meters to 100 meters north.
Roger that. Thank you, Jason.
Pros. Wow.
Cool.
Okay, so we will stop now because there is some iceberg approaching. We are going to move a couple of 100 meters and then try again.
Everybody tell me he can move which way he wants.
Yeah.
Bridge, RV.
Coming up.
Yeah, go ahead.
Okay, Jason, you can move whichever way you want. We'll kind of follow along and then let us know when you're happy you're safe.
Yeah, I'll just function a 50 meter move north towards the following track line.
50 meters north,
Just
okay.
double fine, that'll get clear.
Okay.
The area here is relatively flat, so we assume that there will be quite
RV bridge on the move.
similar
Copy that.
once we move a couple of 100 meters, that we will find same similar biodiversity and also same amount of animals. There's a small shrimp. And it's amazing how much is in the water on little particles. This is all coming from the surface waters, from the water column. This is produced by the plankton. So these are the remains of algae and also small organisms that are settling down to the deep areas here, to the deep sea.
The team that are working with the pelagic sensors are surprised by just how productive these surface waters are. So you can just see in the water that there's a huge amount of material that's raining down and providing energy for this community, basically. I've had a pelagic camera out there and you can just see things drifting and floating and swimming through the water. So despite being quite an extreme environment, it's an incredibly productive one. There is a lot of life here, and even though these open sandy seabeds look quite sparse, you know, your eye is drawn to these large animals every now and then, the sediment itself is teeming with life. So sometimes we think that we get to the rocks and the outcrops and it's suddenly full of life, but that's because the life has no choice but to be on the surface. These are just as teeming with life, but because it's buried and it's much smaller, it can just look like this open desert, but it's actually really, really productive.
We see in the water some jellyfish. These are medusas. Small reddish ones. Here we see another one. And also there are some corals with a large stalk. It's probably related to a coral that we call umbellula. There's also some starfish. These corals are filter feeders, so they found a lot of food here. Oh, now we see it's gone. It was kind of a crustacean swimming in the water. Let's see if we see a clear view. Again, these deep sea sediments are very, very soft. So a very little disturbance brings them in suspension into the water. And then we need to wait a little bit until we get a clear view. We continue transiting to get a little bit more far away from the icebergs that are coming closer. So we are in permanent contact with the bridge that they are advising us to proceed or to stop.
I think the live feed has the radar as well, so they can see what we're up against.
Ah, great.
Are we looking for anything particular with these push cores?
It's just a bulk sample to characterise this area. So the little fella down there is an eel pout. We've got a couple of these now, and this is their defence, is to coil up and pretend not to be a fish, which made them very, very easy to catch. We've caught 2 of these so far. We're trying to figure out how that makes sense when this environment is sort of as it usually is, because this is really unusual to have the lights on and to see these animals in this way. So we're trying to imagine this in complete darkness with just the bioluminescence in the water to sort of pick out the outline of things. So maybe when they're disturbed and they curl up like that, I don't know, maybe it looks like a jelly and something you wouldn't want to take a bite out of. Or maybe you're just making yourself as small a target as possible and staying still so a predator can't find you based on vibration. But unfortunately, the new apex predator, the ROV, isn't fooled by such thing. So the poor little guys have been fairly easy to catch.
So it's the first time we see them, but also the first time they see us. So whenever the bridge is happy, we could then stop.
Bridge, RV.
Yeah, RV bridge.
How is this location looking for it, Jason?
Yeah, that's good. We've just come to a stop now. So yes, I think it's good to go.
Awesome, appreciate it. Okay.
Okay, then we are preparing for landing again.
Let's
We see a small sea urchin, the red one, and a small sea anemone, the white one.
Alright, that's what's on the bottom.
So there are different types of bush corals. We have some large ones for the geology and the standard ones. And we will begin with the standard bush corals. We can put in total 12 of them. Exactly, and for this, maybe a little bit more. Yeah, that's fine. So we want to leave a little bit of water at the top,
What's coming out?
so that the sediment will not flow out of the coral at the top.
Teresa wants her 3 push cores here as well, so it's all the push cores here. So that's easy.
All 3.
Yes, because she's seen the species after in the area, so it doesn't have to be right next to them.
Okay. The first time I came here to Antarctica, it was in the year 2001. We went to Elephant Island and King George Island with a German icebreaker. It's called Polarstern. And at that time we didn't have ROVs, and we didn't have these possibilities. So we are taking samples more or less blind. We didn't know where the gears were landing, and they were all mechanic gears that just are designed to take samples from the sediment or from the water. But now we are very happy that we have this possibility to use the ROV. It is always fantastic to see how the seafloor is looking like. This is changing our view of the oceans. You need to know that you can have remote sensing on land. So from satellites, you can get a lot of information. You can get also photographs. But the ocean is preventing that we can really look very deep into the seafloor, the water. We have remote sensing. We have information from satellites coming from surface waters, but not from the deep sea.
Recovery mode and back or something?
We said you should be fine. Let me just look at
You do it? Or just keep it?
Just keep back. I didn't do that, but I'll keep an eye on
All
it.
right.
Trying to pull the.
A couple of people in chat asking about using different light sources. Yeah, there's been a few different light experiments we've played with. The equipment I've got here with me for the pelagic stuff has far red light to try and not disturb the animals, but it's just absorbed so quickly by the water. UV's a fun one. Black light is a fun one to use as well, because certain compounds fluoresce, and ones that really tend to are the bioluminescent pigments. So if you've got an animal you think might bioluminesce, then shining it with a black light, you can sort of pick out all those photophores on the side of them. So it's handy for things like myctophids, where you've got to count them and figure out where they all are to try and see what species you've got. It's hard to deploy in the field, but it is something we've explored, and it sort of reveals a whole extra world. Things are so shaped by the water and the light that it absorbs, and also the light that's produced by the animals, that they sort of exist in a very different photic realm than us, and it's something we need to bear in mind when we study these animals, that they don't see things the way that we see them.
Also, there has been some tests with multispectral cameras. This means that the light is decomposed in the different wavelengths and then saved as a picture in different wavelengths. So you can then analyze the reflection or the emission in the different wavelengths. Some of them are proxies for instance, chlorophyll, so it's a proxy for primary production, or other kinds of chemical compounds. But this is really something that is starting now. It's not something that is regularly used in all the ROV dives. And also the 3D vision, Tom. You are also starting to play a little bit with 3D video.
Yeah, my system has a 360 camera on it, because we're working with the sort of pelagic realm, and there's a few different things to overcome there, and one of them is that it's just this boundless 3D space in all directions. So we've tried to set up a 360 camera so that basically nothing can sneak up on us, we don't miss things, and it's already paying off actually. I've got a deeper water camera that faces in one direction and a 360 camera on a shallower rig, and we see a lot more things that we would have missed. You've essentially doubled your field of view. Plus it's cool, I can put it on a Google Cardboard and you can pretend you're down there, which is good fun.
There are some black light. We have tried this kind of illumination in shallow waters, in coral reefs, for instance. Yeah, because there are some species that they emit some light when they are illuminated with blue light or ultraviolet light, and you can also make some inference about how healthy the corals are, which parts of them are alive, which parts of them are dead, or they have some illness. But in the deep sea, I have not tried them actually. It would be interesting to use them.
There's a reason that most of the bioluminescence is in about the sort of 450, 480 nanometer range, which is this nice sort of greeny blue, because that's what penetrates the water best. And that's a huge generalization depending on the pigments in the water, with the chlorophyll and things like that. It does actually vary, but that is the sweet spot that the animals tend to use. Then there are some sneaky animals who deliberately use a different wavelength, knowing that their prey won't be able to see it. Things like the stoplight loose jaw. But that is the sort of best wavelength for if you just want to see as far as possible. So things like infrared, which I have on my camera system, but just gets drunk by the seawater basically. It doesn't penetrate very far at all. These different sort of energy levels on the wavelengths just get absorbed at different amounts.
And how much do you think, Tom, the light emitted by bioluminescent fish, for instance, how long you think it can reach, or it can detect it by other animals? It is something very close, or?
I think a lot of them, well, it sort of depends on both the transmitter and the receiver. So some fish just opt out and have gone for different sensors, and that's when you get some that have totally removed their eyes, essentially, things like the faceless cusk eel. As juveniles, they look like regular fish, but as they go deeper and they specialize, their skin actually grows over their eyes, and they end up with a a kind of creepy blank looking face. If you see pictures of them, the things that you think are eyes are actually the nostrils, and it's got no eyes at all. So that's a good Google hit for you. The faceless cusk eel, it's worth having a look at one of those, they're weird. Some then specialise the other way and get big collectors, but even some of those, we know from their brains that vision isn't their primary sense, so it's kind of like thinking like a massive satellite dish to pick up a weak signal, but it's maybe not even their primary sense, even though some of these have huge eyes, and that's all we can notice when we look at their brains, that they're not actually doing a great deal with it. I think a lot of them almost have a really slow shutter speed, so they're not resolving things quickly like we do. They're kind of building up a heat map of where the light is, so they would see like these vapor trails, and they would see an image slowly build up over time, and sort of come out from the background. It's worth remembering as well that there's bioluminescent bacteria in the water, so even just moving, you are picking out your silhouette essentially. So that's why, especially up in the pelagic realm, you've got to be really sneaky and subtle, and I think some of these body forms are about not disturbing the water as much as they're about conserving energy.
Oh, that's very nice. I like this idea. So this is like a long time exposure. When we photograph the sky, which has also very little light, and you let a long exposure, then you can see at the end a lot of stars and so on. So you think that they use the same approach, that they kind of incorporate or add the light over several minutes or whatever?
Yes, yeah, I think so. I think it's sort of it's constantly sort of refreshing and building up. So it's not a quick reaction. It's a sort of general background. This is in some of them. The one I always thought about was maybe the Bathysaurus, the big predators that sit and wait on the bottom. They've got quite large eyes, but they're in a very low light environment, and they're probably not looking for things advertising their presence, but things that are just disturbing the general background bioluminescence.
And also it was reported that some shrimp that they are living in these hydrothermal vents, that they could probably also detect infrared light as a temperature sensor, let's say, in order for them to be more easy to locate these hot vents. They have some organ in the head that they it seems that they are able to detect red or infrared light.
And that thing's weird, isn't it? It's like a solar panel. It's right on the back of the carapace, sort of behind the eyes. They've got this huge collecting organ. So it's not like they've adapted their eyes. This is a totally novel structure.
Organ, yes.
And they don't have traditional eyes, that species, is that right? I know some of them have eyes, and then some of them have this sensor, and then they don't have eyes.
Yeah, I think they have reduced the eyes, yeah, that's true. So there is a sea star there that we didn't notice, but it's good not to hit the sea star. Now you have it. Can you put it in another place, the coral?
What's that?
You hit a sea star. We didn't notice that there is a sea star there.
Okay.
Just move the coral to another place. Yeah.
Okay.
Otherwise, we have a piece of an arm in the coral, in the sediment core. Yeah, that's a good point. So there are 12 cores for biology, and as far as I understood, Teresa also wants 3 cores here, right?
Yes, yeah. Teresa says you can take hers here because you've seen the species.
Okay. Look, Tom, there is a fish in the water.
There we go. That's doing the party trick.
We don't see you.
I'm a jellyfish.
Yeah. That's amazing. So it's really, yeah.
Yeah. It's a type of eel pout. There's some pelagic members of this genus as well, and apparently they do the same thing. So multiple fish have had this idea, but it doesn't work very well with ROVs because you could just waft them directly into a net.
So let's see, now we need to concentrate a little bit how many corals we have.
That's number 12.
That's number
Right
12.
here. So after this, I'm going to put all these back, and then I'll get the ones that are more deep.
Yeah,
You know?
that's good. So, Tom, what we can do is to get these 12 corals here, and then move a little bit.
Yeah.
When we see a holothurian, we stop, and then we take the holothurian and the corals.
I think Teresa said because the holothurians have been confirmed in the area,
Yeah.
then we can take them. We can just take them at the same time, so we don't have to be so specifically next to the holothurian.
Mhm.
But we do have to make sure we grab them later.
Yeah. I just say because now we have really disturbed a little bit the area here with our 12 push corals. Maybe I would prefer to just move a little bit, and in the next stop we take the other corals. So now we are recovering the push corals. See, yeah, everything went well. We analyzed the top 5 centimeters of the sediment. This is because in these top 5 centimeters, most of the fauna is living there. There are 2 reasons for that. The food is coming from the water column, so it's accumulating at the surface of the sediment, and this is where the animals want to be. This is where they get the energy to survive. And the other point is that the deeper you go in the sediment, the less oxygen there is. We have made some measurements, so we think that the first 5, 6, 7 centimeters they have enough oxygen, but then it starts decreasing, and many organisms they don't like when it is getting with less oxygen. Of course, there are some other organisms, some bacteria, also some worms, that they like these anoxic conditions or low oxygen conditions. But they are not everywhere. So we sample, then we extract from these corals the top 5 centimeters, and then we will analyze how much fauna is living there and which species are living there, how much is the biodiversity. So there was a question in the chat asking for what program you are specifically taking these samples. We are characterizing this area of the Antarctica, the Bellinghausen Sea, which is an area that has been studied very, very little. And there are several questions that we are trying to understand. First is, of course, how much biodiversity, how many species are living here. Are the species known? Are the species the same ones that we know from other places in Antarctica? And also we are comparing with the southern part of Chile. Then we are collecting in areas that has been covered by ice during the past decades, but now they are ice-free. And we want to understand how is the colonization of these areas by the different organisms. This part of the Antarctica is affected by the warming of the waters more than other areas in the Antarctic continent.
Pedro, should we push them a little bit deeper before we collect? Some of them quite low.
Okay. So the colleagues in the chat are asking us to push them a little bit deeper. So before you take it, if you can push it, let's say 2 centimeters deeper. For instance, the 16 is quite, yeah. Okay, go. A little bit, yeah, stop. Something like that would be good. The deeper we take the coral, the more safe it is, so the sediments will not fall down from the coral when we take them. But again, we are, for this specific corals, we are interested in having a little bit of water on the top so that we are sure that the sediment surface is not disturbed. Here we see the sea star having a siesta, laying on the bush corals. Some of the sea stars here, they get huge, they get really very big.
There's that giant orange one with the black tips on the arms. I haven't learnt the name of it yet, but
Yeah.
it's like a large house cat.
Yeah, exactly. We got the name actually. Let's see if I remember. I mean, I had it in the. And these they get really huge, really. There are two reasons why animals get big. Either they get a lot of food. This may be a reason here, but also it may be that they are very old. So they keep growing, and if they are not disturbed, and I mean in Antarctica there is not a lot of human activities. So it may be that these organisms, they are old. We have little information on how old the benthic animals get. We tend to believe that corals get very old and sponges also get very old. We are talking about hundreds of years. Huh? But how old is sea star? This is a very good question. There has been some dating of brittle stars related with the sea stars because they are also accumulating in some skeleton fragments. They are accumulating like growing rings. And to our surprise, some of the sea stars that we had collected in the deep sea, they were also 25 and 30 years old. So I assume that many of the organisms that we see here, they can also be quite old. The age of the animals and the growing rate and reproduction of the animals, this is making a huge difference for the ability to recolonize or to recover from a disturbance. Of course, animals are growing very slow and get very old, and they reproduce only once in a couple of years. Then it will take a long time if we disturb the habitat or if the climate changes the habitat until they will recover or recolonize other areas.
With the amount of productivity that we're seeing, I'm surprised we're not getting an anoxic layer. This sediment's golden all the way down.
Yeah, then maybe I think in one of the first dives, I think it was the first dive, I think they had some suboxic sediments in about 6 centimeter depth.
I've only seen little patches that I thought was something that maybe died and then just went anoxic locally. It's interesting how well oxygenated it is.
There is a question in the chat. I mentioned that specifically the Bellinghausen Sea, this area is more affected by global warming than other areas in the Antarctic continent. This is because here warmer waters from the north, from South America, are coming closer and they are almost hitting the Antarctic shelf. And this provokes that the shelf ice is melting quicker. And this is the question is how this is affecting to the biodiversity. And this is exactly what we want to know. This is one of the first times that samples have been taken here. I mean, there's a lot of studies on, for instance, the penguin colonies and other mammals and terrestrial. There has been also some benthic sampling, but what we want to do now, it's kind of a baseline to know how the state of the biomass and biodiversity in this area now, and we will then probably, we or other scientists will follow up in future with follow-up cruises and see how this is changing. We have an area where a big piece of ice, of shelf ice, has broken up a couple of weeks ago. We are planning to visit this area, and we will be very lucky because this will be kind of the time zero after the ice is breaking, and then in the next years ourselves or other scientists can come here and control how it is evolving. All this information that we gain here is going to be published in international journals that are peer-reviewed, and this information is available to other scientists. And so it is actually not needed that we come back, other people can come back and continue the studies.
Are we getting the geology course in this lot as well?
We move very
We
quickly.
move for the... Okay, cool. It's kind of... Yeah. I'm running out of space.
Yeah. Are we going to do some microbiology tests? I'm not really sure. Tom, do you know if anybody's doing microbiology on that?
Not specifically on this trip. It's a whole other realm, that one.
So it seems that we don't have any microbiologists on board of this cruise. Samples will be taken home. We have samples that are fixed in ethanol, and others will be, I think they will be frozen.
It's difficult with export, isn't it? You've got to be...
Yeah.
Because all of our specimens have to be non-viable, whereas if you do microbiology, you need it alive.
Yeah. So that they will be available for other studies. So there is some measurements of biological activities and also of proteins in the sediment. This is done by Teresa. So there is a biochemical characterization of the sediments. Specifically bacterial biodiversity. I don't think that we have anybody on board, but with the samples it could be done. If some scientist is interested in that in the future, they can contact us, and probably it will be easy to be done.
Some of the chat talking about how unexplored this area is, and I didn't realize just how unexplored it was. So a lot of our maps of the ocean are gravity derived. So we use satellites and look at the changes that have been made to the sea surface by the gravity of the rock under it, essentially. And we don't even have those for here because it's usually covered under ice. So there is nothing known from this area. We don't even have these very broad stroke maps. So we've got some very exciting mapping done as well. I know the geology team were very excited last night with all these interesting structures that are being revealed and what they tell us about what the ice is doing and what the ice has been historically doing as well. That's the most interesting thing. It leaves these scars in the seabed that they can sort of back calculate and figure out just what's been going on here at the sort of millennia scale.
Many areas in Antarctica are not explored. I mean, there are some places where we have these Antarctic bases, that they use for also terrestrial exploration, but also overwintering and meteorological exploration. Okay, wait a minute. So we have collected the 12 push scores for biology, and because everything is a little bit disturbed, I would suggest that we move a little bit When we see a nice sea urchin, we will stop. Then we will collect the sea urchin and take the additional chorus for the geology.
Right.
So, sea cucumber.
Ah, sorry. I said sea urchin. This is not right. We are looking for sea cucumbers.
I have a wanted poster in front of me. I'm not very good with my sea cucumbers. This one with the frilly bits.
Yeah. So it's,
Yeah, we'll probably go like 20 meters, I think, right?
yeah, it's good if we move a little bit.
Yeah, at least that. Yeah.
Go ahead, RV.
Updated waypoint.
Copy it.
That cloud will follow us a bit more.
Is that one of them?
Beniagone is a holothurian that, or sea urchin that is also able to swim. The ones that we see here, they are, yeah, I think maybe you're right. This is one of the, that's a Beniagone, and the other one with the larger tentacles, let's say, is probably Scotoplanus. But I mean, this is a good spot, I think.
Are these not Teresa's target species here?
Exactly, yes, these are
Yeah.
both of them. So, to the ROV pilots, we are targeting this kind of organisms, so this these holothurians, sea cucumbers, and whenever you see, they are everywhere. So I mean, we can move a little bit, but whenever you think it's a good place to stop, we can stop and collect some of them.
Okay. Those guys up
Yeah,
there.
exactly. So it seems that they are very common here, very abundant.
And she wants it softly with the scoop.
With the scoop.
With the scoop.
In the bio box.
Yeah. And I think they can share a bio box. Right.
If this is, yeah, exactly. If this is the same species, we can put them in the same bio box.
So I think that chunky fella at the front is the type she's after.
Okay. Well, that's
You mean this
a
one?
chunky fella.
I thought so.
Yeah.
This
Yes.
is the one here?
Yeah.
Let's grab
Is that a big star in the back there?
it.
Yeah.
So we have two species of holothurians here. And let me confirm what of them she wants to. Oh, this is a very nice view. Very nice. So, again, we are looking. We have here a identification book. Protoelpidia or Scotoplanus. These are two candidates.
Oh,
For the
she's
name?
saying that one.
Yeah, that one. Perfect.
Scoopy Scoopy.
So they are there, holothurian for about 20 minutes, and then holothurian for about 20 minutes, and then view this movie in fast forward. We will see that they are really active and they are moving and putting material into the mouth and also moving slowly with the little legs that they have. So it's a different speed that in the universe than our speed. They are moving very slowly. If you take notice it, you will notice that the tentacles around the mouth, the very little ones, they move very slowly.
We'll scoop this guy into the bottom box. And then we're going to do the rest of the push cores. She wants it to scoop. It's very soft, apparently. I think she just doesn't trust it. That's fine. Seems like the right decision there.
In other parts of the ocean, these holothurians, these sea cucumbers, or specifically these ones, what are on the sediment, they are also called sea pigs. Well, in my opinion, it would be much better to call them sea cows, because they look like cows that are on the field and grazing. And in other parts of the ocean, like the Porcupine Abyssal Plain, southern of UK, they have suddenly massive explosion of their abundance. So they get very, very, very abundant on the deep sea. And we don't know exactly why this happens. If this is a kind of, it's not seasonality because it doesn't happen every year. And there has been a lot of studies following this, specifically the event which is known as the Amperima event, because the species was Amperima rosea, how this explosion in the abundance of the Amperima affected the biodiversity of other animals. And it was clear that after the Amperima event, some of the organisms, they also increased their abundance, but other organisms, they actually didn't care a lot.
Oh, this is surgical. This is beautiful.
Yeah, it is. Here we go.
We did go and pick a big one, didn't we?
Mm-hmm. We see also some brittle stars close to it. The brittle stars are very common in deep sea waters. Here you also saw very nice the brownish sediment at the top, and then more pale sediment at the more inside at the bottom. You see this brownish sediment. There is much more organic material. This is what is very interesting for life, for other organisms like worms or crustaceans or little meiofauna organisms, water bears, mites.
Biobox 4 Pedro, or a different one?
Excuse me?
Biobox 4?
Biobox 4.
Does that work?
Yeah. Biobox 4, we need to document everything very accurately, because then this dive will last for several hours, so we need to know in which place we have put which animal, that we can trace back the depth, and the different parameters also. So this is going BB4A.
That's the goal, right?
Can you zoom in one second?
You got it, buddy. So coming in. Yeah, let me try to clear some of that sediment out for you.
I think the. Well, we focused in on it for a long time, because it's every 10 seconds. So we'll have a few then.
Yep.
So start to go.
Oh, okay, it went to Biobox 4D.
That's what we wanted.
Yeah, that's what we
Very
said,
good.
right?
This feels like a carnival game.
Yeah, very good. So this is also the reason that we write down the place once we are sure that it is there. It may also happen that the animal falls down and gets out of the box or something like this.
We don't count our chickens until the door's closed.
So what we can do next is to take the 3 normal push coras and the geology push coras, but they're a little bit larger.
And that includes the long ones.
The long ones, I mean, yeah.
So, which one are we taking?
So we got all the front row. Yeah. So 3 and 3, so we're only doing 6 here?
Yes.
Okay, I'm gonna lay this.
This includes 3 which are longer.
Do you want to move somewhere else? Do you want to move a little bit further forward for better visibility?
Yeah, we may do that, yes.
That's
Yeah. If you prefer that, it's good because, again, the sediments are very, very fine. And once we start touching the sediment, it gets everything in suspension. And it gets a long time to settle down. In total, we will want to collect 3 of these holothurians. So we have 1, and later on we will try to find 2 additional ones of the same species. But what we could do is first to take the push coras. We land somewhere, not very far, and we take the push coras then.
Let's go up a little bit.
There is a question in the chat if we think that the solar maximum have an effect on global warming. I am not a meteorologist or oceanographer, let's say. I'm a biologist. But my understanding is that, of course, the different phases of the sun, there are periods where the sun is closer to the earth and periods that is more far away, that this is of course affecting the climate on earth, and it is, when it's closer, it's also affecting global warming. The general idea, however, is that we humans are helping or accelerating, let's say, the warming of the atmosphere, and this is repercuting in the warming of the waters by producing greenhouse gases and putting them in the atmosphere. This is a beautiful view.
Yeah.
Look how many different. Maybe we can take a photo here.
There's another
We
2
have
of
a-
our target species as well.
Yes.
And a little ice fish chilling out over there.
You want to land
work
here,
to-
Tom?
I just noticed that there's a couple more of the target holothurians.
Yeah.
There's a little pogo, little plunderfish over by that
So set down here,
sea pen.
Yep.
do the cores, and we can grab those 2. Alright.
So in general, we are coming, going out of the last glaciation. This means that the Earth is getting warmer in a natural way, so it's something natural. But humans are kind of accelerating this process, and this is what we worry about. Well, the shifting of the poles, I think that you mean the magnetic pole. I don't know if this have an effect on global warming. I would tend to believe that not, but again, I'm not an expert in this field. What holothurians eat? This is specifically something that we want to know. Of course, we think that they are deposit feeders, but other selective feeders or they are not selective feeders, or they are eating everything that is at the bottom, or they are just picking up some of the algae or some of the available food. And in order to do that, this is the reason but we are also collecting the holothurians to do some lipid analysis in the tissue in order to know which algae they are feeding on, and in order to study what is the contribution of the holothurians in the food web.
That's why we're being so gentle when we collect them, because we don't want them to poop.
Yes.
Which is the first thing they do when they get stressed.
This is a beautiful place. So yeah, 3 normal push corals and the 3 long push corals is what we are going to take here.
And these are all geology, aren't they?
Yeah.
So
Yeah.
these can go right down to the top.
Yes, exactly. All 3 are for geology, so they don't want to have a lot of water at the top. They prefer to have longer corals because they are looking into the stratification, they are looking into the sediment. For them, the longer the coral, the sediment in the coral, the better. Weather and so climate and temperature.
They were on 6 though. Yeah. True. Yeah, whichever ones. Is this enough porch? Do you want some more?
So wait,
Good.
okay. We need to concentrate a little bit now on the push corals.
Is there any icebergs?
No, we should be
There.
here,
Are you racked out?
yeah.
The long corals, they have some tape so that we can identify them better. And this can go as deep as possible. Of course, without the sediment coming out on the top, but so that you don't see any water anymore. Yeah, I think that will be good. Yeah. Yeah, excellent. Very good.
Short one?
Yeah, we,
5?
yeah, it's a short one. Can do 8 if you want. That's easy to get up.
8.
Yeah. This guy over there, right there.
There is a question in the chat. They say that they cannot see the overlay of the, let's say, the depth and other information.
It should be. Yeah, I was throwing a bunch of thoughts.
Once we've got all the cores in, we can grab the cucumber before we pull.
Yeah, it's off. Alright, if you do 4 and 3 then that'll be the whole shebang. Whole shebang. One looks good.
Here?
Yeah.
Thank you.
Yeah.
Here?
Yeah. So still good.
The sediments here, they are very soft because most of the sediment is the accumulation of the remains that are coming from the plankton, from the surface waters. Of course, the icebergs, they are also transporting sediments and rocks. So we have these drop stones. But here we have this accumulation of very, very small particles, and this takes some time as it's slowly accumulating and building these very soft sediments on the top. Of course, deeper into the sediment it gets more compacted, but this is not because this could, yeah, this exactly. So this could go a little bit deeper if possible. It's not because of the hydrostatic pressure, but because of the weight of the sediment itself that it starts compacting and dewatering it. So it's getting less water inside and it's getting more compacted.
It's a strange thing to come to terms with when we're thinking about the pressure, because it's such an overriding thought when we're at these depths, but it's sort of equilibrated basically. So even these holothurians, the sea cucumbers, they're soft and squishy in your hand, but the pressure is equalized inside and outside of them.
You can grab them?
Yes, we can grab them now.
Did you want to grab the holothurian first?
Before we
Before
get
we stir it
the,
up?
what would
Can
be
we
your
reach that far?
advice? To get the holothurian first or first the push cores?
No, at this point we have to grab it first. Oh, this one?
Yeah.
Okay, I could grab this, and where do you want to put this?
In the standby box.
Yeah.
I'm gonna grab this instead of using a scooper, right?
So let's use the other arm.
Okay.
to light the bench.
I think she wants them collected very gently. We basically need them not to poop.
Okay.
So
It won't poop.
she's requested to put the scoop.
Scoop. Can you reach for the
And,
scoop though, Ben?
no, we're gonna grab this we're gonna grab the push push first.
Okay.
Okay.
Then we will get the
Make a
push
mental
cores
note
back,
where he is.
and then
They don't move very fast, right? So
no, and then we get the holothurian.
So, where's 8? No, I'm gonna grab 5 first.
Okay. So 5 is a short one. Good. Like that.
science catalog.
Oh, yeah. I have to use the sick hand with the force now.
Can you zoom in and see?
I can.
Go down a bit.
Down.
There.
So if you have incorporated just to the dive, we are in the Bellinghausen Sea. We are in about 1,000 meters depth. This is one of the deepest places that we have found here near the ice shelf. And we have proceeded to collect some sediment using the push cores. Some of the cores will be for biology, and now we are recovering additional push cores that are being investigated for geology and biochemistry of the sediments. Then we collected also they call them again a sea pig, holothurian, and at the moment we are recovering the push cores, and then we will proceed to collect 2 additional sea cucumbers. We expect to find here a lot of new species, specifically the smaller organisms, the worms, the crustaceans that are in the sediment, like copperpods, and different types of worms, the mollusks, like scallops and bivalves. The other point is that even if the species are already described, because some of the species, they are, we call it circum-Antarctic, so they are in different places across the Antarctic shelf. But there is very little studies on the genetics of these organisms, so how these populations are connected. This is also a question that is very interesting for us, to know how the different places in Antarctica are connected, because if different places are affected differently by global warming. One place could serve as a nursery place or a recolonization place for other areas. So we don't know exactly how the different basins and the different areas, the different seas are connected. What we know, of course, is that there is a circum-Antarctic current. There is a current going all around the Antarctica and isolating the continent from the southern parts of the ocean, the Pacific, the Atlantic, the Indian Ocean. But how exactly they are the different areas in the Antarctica connected is not very well studied. Now push core number 4 or 3, it depends. In the background we see in the water some swimming holothurians, Peniagone. At the moment there is, in the top right, there is a jellyfish drifting slowly. The jellyfish, they belong to what we call, when they are very small, they belong to the plankton, so they cannot swim against the currents. And there are other organisms that they can decide where to go, like fish or shrimps or other organisms. But if the organisms are very small, they are drifted with the current. So this is what we call plankton. We have some, I see there is kind of also a rolled fish over there. Tom, can you confirm?
It's looking fishy. I'm not sure what it is though.
Yeah. Okay, now we have the last bush coral to be recovered.
Is that our complete set? I lost track of it.
Yes, this would be then the complete set. That's if the pilots can confirm to us that yes, we have used all of them. Let me check. So we had the 12 course for biology, and here we had 3 long ones and 3 small ones. I think there is one missing.
And that's everything, isn't it? So I'm not sure we've used some of the ones in the front.
Okay. I think that we should have 2 empty push cores. I think that we I think that we need
One and two.
2 and 2. But first we can. Yeah, exactly. So this, I'm sorry, we forgot these two, but we should take them also here. Whatever you prefer, to take the holothurian first, and then we take 2 additional push coros, or the opposite.
You want to take 1 and 2 here as well?
Yes, there is one. This one.
Yeah, you went to the push course?
Yes, exactly. Exactly. These 2 push coros, we need to take them also here. Sorry,
Okay.
we thought that we were done, but we have these 2 additional.
I'm gonna use the
Whatever is more convenient to you.
squeeze this.
There's going to be a lot of processing later.
Yes, this is going to be a long dive, and after the dive, we need to process all these samples as deep as possible. Yeah, I think that's good.
Try and avoid clipping that cucumber. Just now helpfully close. Oh, lovely question in the chat. Someone's asking me if there's a fish I'd really like to see in situ that we haven't before. There are loads of them. So many of the deep sea fish are only known from very badly damaged specimens, and we have to sort of back calculate what they do. The example I always give is the tubai, that one with the sometimes called a spookfish, the one with the big transparent head, and its eyes are actually inside looking through that like a fighter jet cockpit. We knew about those since the 1950s, but we didn't know about that clear dome until they were filmed with ROV, because so many of these animals are covered in this fragile gelatinous layer, or these long filamentous fins that just get destroyed on the way up, and then we have to try and figure out how on earth these work in life. So things like that, things that we've maybe known about for a long time, but then they live in a totally different way once we see them alive. Personal one for me, I love the snailfish, and I reckon there's parental care or some sort of brooding in the Hadal Trench snailfish, and I'd love to find out where they're laying their eggs and looking after them.
with the gravity core, we can take long sediment cores. We are also using the gravity core on this cruise. Then you have from a small spot a core that maybe what we had the last days, it was about 2 meters. In some other areas we can have gravity cores with up to 5 to 6 meters depth or even longer. And then you can get the paleo record of the sediments and analyze the biochemistry and the different parameters in different sediment strata that belong to different ages. What we can do with the ROV is to take many different, longer amount of sediment cores for different purposes, for biology, for geochemistry, for sediment granulometry, all these kind of things. And we are more interested in the surface sediments, so therefore the cores here are short because we are processing just the top surface sediments.
Holothurian takeoff.
Yeah, Peniagone is flying. It's wonderful. Look at this. We didn't know that these holothurians are able to swim until we got the first cameras into the deep sea. This was more or less in the 80s. In the 80s that they were, it was clear, some footage appeared that these holothurians can also move swimming, and they can also displace for several kilometers, drifting, swimming but drifting in the water. And here we see a huge copepod coming into the field. Copepods are normally small, but this is a couple of centimeters big. And now it's drifting out on the left side.
That's some very nice piloting to take that core out with barely disturbing the holothurian.
So perfect. Now we are done with the push cores. The next target would be to collect 2 holothurians.
And there's 2 in shot if we're in a good spot.
We are, yeah, it could be that one that it will be reachable by the ROV arm. And the other one is probably too far away where we need to move.
Well done.
So 2 sort of sea pigs that are in shot now, or after with the scoop. I don't know if it's worth shifting the body around. Whichever's easier.
Right. You gotta pick that one up. You wanna pick that one?
Yeah, yeah. Well, it's going to one of the same. Side skills.
You ready?
Yeah.
Good?
Yeah.
You gotta pick up the scoop. Scoops.
Scoops and
Scoops
screw
are
around.
and then we're gonna put that here.
Seabird.
Cucumber.
So we are changing pilots now. We'll make a little break and we will continue in a few seconds, minutes. Now, here we go. The ROV has 2 arms, one on the left, one on the right. And we need to see with which arm it's more easy to grab things or to take the chorus and so on. One of the arms is also more, can do more pressure than the other. But in general, the decision is just to see which of them is more easy to use according to the target.
The pilots have a huge amount they have to juggle when operating this. So you've got to position the vehicle, but at the same time you don't want to reverse, you don't want to stir up all the sediment and lose your vision. And then the manipulators both have a range of motion, and you almost sort of lock the elbow when you sort of try and come in. So there's an arm on each side. Each has sort of a swath of area that it can work with, but then they're specialised as well. Like Pedro was saying, the one on the left has a softer grip, and so we can pick things up a little bit more gently and with a bit more of a nuanced grip. And then the one on the right is the beefy one. It's a little bit stronger and handles tools a little bit better because it doesn't have that soft grip. So you'll see when the arm comes into view, there's actually grooves and notches all in that pincer, and they fit the different sort of tools. The standard sort of ROV interface is what we call a T-bar, and it sits really nicely into these grooves in the handle. So when the claw is closed, you're essentially, the tool you're using is an extension of the arm. You don't have to sort of wobble it around and move it independently. So you can see that with this scoop here. We have extra challenges right now because there's 2 vehicles in the water, so it's always really complicated. The vessel is trying to keep up with us at the surface. We're getting all distracted and all excited about what's going on down here. And up above us, they're dodging icebergs and ice flows. So every now and then we get the call that we're just going to have to pack up and move and follow the ship because the ship has to do some dodging.
Where do you want him to go?
Has a friend in by a box 4D,
So maybe one of the other compartments.
can they share?
Yeah.
Yeah, so that it could go in 4D again.
D or B?
D, Delta.
Okay.
Oh, you fell. Sorry.
Aha.
Yeah.
Will he be too disturbed now, or will we try again? Yeah, try again. Is he still in shot?
Unfortunately, the Horosurian fall down. We don't see it at the moment. There is another one a little bit far away. It's not reachable by the arm at the moment.
See him, Teresa?
I think that we could move a little bit if this is more convenient.
There is the one at the base of that C-pan.
Yeah.
Oh, eluded.
Let's see.
Maybe we try to move in order to get more clear view.
They don't seem rare. We might be able to find another sign. Okay, if we move on a little bit so we can see. I'm not sure we. Yeah, but I'm not sure if we've disturbed it so much that it's going to be tricky.
Here we see another penny agony swimming. They're wonderful.
So yeah, that's a target one right in the
Yeah.
center now at the bottom.
Yes.
Mysid, doing cartwheels. Nice.
Yes. Okay, we are landing back.
just in front of it is a little organophrine, I think.
A crinoid, the small one, right?
The plunderfish, the little fish.
Ah, okay, that one, yes.
We'll get the cucumber, but if he stays put, I may have more difficult requests.
So how you would collect that fish?
Might be slurp gun.
Yeah.
They do have a net. Not a good multitasker, and the amount of things that the pilots have to keep in mind, I don't envy.
Yeah, very nice.
Yeah.
No, stay there.
They're not much denser than the water around them, so they're well light and fluffy.
Okay.
I think we just pushed the pogo along like those 2p coin machines at the arcade.
Yeah.
Might still be there.
So now we're trying to put the specimen in the biobox 4D. Here we go. Hopefully it will, yeah, it went in
Nice.
perfectly.
Well done.
Well done, very good. Okay, and then we just need an additional one of those. Probably if we move a little bit, we will find some.
Yeah.
We see always in the water some polychaetes swimming, some worms. There's some jellyfish, there is some mycetes with small crustaceans. We saw also a huge copepod. So it's a lot going on. Yeah, we see again a copepod, a big one. Oops, yeah. Okay. So maybe just in front of us there is one. Let's see. It's a little bit sediment in the water. Maybe we move in order to get a clear view. That's not the species. That's a different species.
That's a different one.
That's a different one.
So chunky boys.
Mhm.
Okay.
So So we continue moving.
In the direction of waypoint 2. See what we see.
Here there is one here. So this could be our next target.
That's the one. Chunky boys.
They are really big.
They are. They don't fit in the scoop. You're gonna need a bigger scoop.
So this is the species that we are targeting. So we have already collected 2 of them, and now we go for the 3rd one.
To complete the set. Teresa's just pointing out the wonderful poop it's done.
Very good.
Excuse, we were talking earlier about whether they're selective feeders or whether they just sort of scoop everything into their mouths, and that's what she's studying. So she wants the sediment before it goes into the animal, and she wants an undisturbed animal that hasn't pooped out what it's eaten, which is quite a tricky thing to do, but
This can
the
go
pilots are
together
nailing it.
in the Biobox 4D, if possible.
4D?
B, same place.
D, delta.
Yes.
Delta. When we recover them at the surface, they look quite nice. They look quite the same shape that they are at the bottom, so they don't change the morphology or deflate or expel water or something like this. So well done. We are at the end of our shift.
Are we?
I need to thank the ROV team, the pilots. They did an excellent work. And I want to thank also the people that are in the chat and looking at our job. Now we need to proceed to waypoint 2. And you will get another set of biologists that will take over and continue exploring the deep waters in the Bellinghausen Sea. Yes, waypoint 2.
Right, shall we?
Go ahead.
Yes, we had an update to like my field.
Copy that. Love you.
Thank you.
It's the only sampling, sediment sampling left with the Sackos.
And stone.
And stones. We didn't see any.
No, we didn't see any rocks, any drop stones. This is something that if we see one, we would like to collect for the geology. But we didn't see any. Probably the sediments are very soft, and if they drop down, they will
Work
go
them.
to the sediment, and we cannot see them. No, it got night. We put the lights on.
We'll try and keep the the geologists happy as well. This is probably the most interdisciplinary cruise I've ever been on. There are a lot of different specialists here, a lot of different fields, and it's actually really nice. We're learning loads from each other. Had some good chats, and it's not just out of interest, but it gives context to the things we're interested in. You know, the geologists are showing how things are sedimented and how things have moved, and then that informs the biology, and then the biology is impacting the geology, and it's all a lovely feedback loop.
There is also quite a lot of sea urchins here. I mean, my impression is that they are more common here than in other places that we have visited. But they are small, they are not very big sea urchins, they are small ones.
What's that?
That one is totally full of nice.
When they're orientated this way, these sea cucumbers trigger my fish sense. They look like fish lying on the bottom because their gut looks like a dorsal fin. I'm just seeing what I want to see. Ooh. Is this rock or sediment?
Yeah, it seems to be a slight depression. We might be going down, right? Yeah, based on the profile, yes. Is it possible just to turn a little bit the RV so we can have a look at the animals around here?
Sure.
Thank you. Just before we continue down.
I caught up with myself.
A lot of crinoids in here.
It's nice. There's a lot going on here.
Okay. Yeah. Okay. Is it possible to collect this rock here? Are you able to park? Yeah. Then yes.
We weren't expecting such an abrupt habitat change, and this is quite a big vehicle. It's the size of a minivan. It's got an umbilical hanging out the back, and we also don't want to stir up the sediment in front of us. So actually maneuvering, changing direction, swinging the body of the machine around, it's a delicate dance if you want to get good shots at the end of it.
Put that in a rock box or where?
On a bio box, please. Yeah.
Where would you like them?
We can put it in by box 1A. Yeah. Can we just have a zoom as well once we pick it up? No, just once we pick it up, it's fine. I know we are.
That one's got the cutter on it, so you don't want to grab rocks with it.
Are those brachiopods on there?
I believe so.
Oh, great.
Yeah.
That'll make a lot of people happy.
Yeah, it's an extra.
Bonus. That's why I've not done
Let me take a zoom on the rock and just have a good look. Thank you. This goes to BB1A if possible.
Okay. Got it?
Mm-hmm.
There's pictures?
Yeah, good. Yeah. Thank you.
Nice.
That's awesome. Wait. Okay. Fantastic.
Is that something fishy right there? What's the stripy thing on the left?
actually put it back on the boat passage. If you can.
Scoop.
Yeah. We won't be using it for a while.
What if I in this skin over here?
Yeah, yeah. We just after the segment. Just waiting to use it.
Just wait till you can see it. Grab it by the skin part.
Not gonna need it anymore.
No, not for a while, no. Thank you. So just before we go, we're gonna also take one of the Niskin bottles. Just before we go.
Yep. Okay, Miskin one.
Mm-hmm.
RB Green, you calling?
Oh, never mind. Have the mic open.
Oh, okay, no worries.
Okay. Word now. It's Miskin one.
Thank you.
As we go down this cliff, you want to be watching the cliff face. Right?
Let's see how deep it is. Yes, we go for a little bit, but then if it's for a long time, we just turn it back around, and then just fly out until it reaches 4 meters. Yeah, we have seen this one. Yeah, the pinkish one. We can. Meters, yeah, then it's fine. Trying to have a good look at the wall. Can you zoom out, if possible? Thank you. Yeah, fantastic. Yeah, it's incredible. It's full of echinoderms and yeah, some crinoids, some of the sea stars, some sponges, a lot of brachiopods all around. And I believe that's a coral. Can we stop and just have a quick look as well? Just here in the corner. Just to zoom in. No, it's hydrozoans, the colonial hydrozoans. Okay, thank you. This seems like it's correct then? Anna? Huge Brazen sea star. Is it possible to just back just a little bit so they're not so close? I don't know. Yeah. Perfect, thank you. Yeah. This way maybe we can see a bit more, I'm not sure. We have a lot of sediment in suspension all around. Thank you. Thank you so much. Fantastic. Can go back to the waypoint. And until waypoint 3, we just go in transect mode, so just flying around and have a look. That was a beautiful rock. I like to try to fly about a meter from the seafloor. Mm-hmm.
Yeah, I understand. Yeah, meter.
Yeah, perfect. Mm-hmm. Is it possible to dim just a little bit the upper right? No, not possible. Yeah. Can try. Yeah, we have way too much organic matter in suspension here.
Yeah.
point, getting a swing.
Okay, that's all right. Thank you. Yeah, it's cool. Yeah, it's what it is. A lot of organic matter, and you know, it's what we have. The animals love it. That's why they're here. So it's perfect. Cool. And
What do you mean you can't open chocolate bars? Hi, Sophia.
hi, Sophia. Double Sophia.
Hang on, more wanted.
Oh.
Tea, Sophia's school.
But I don't need two.
No, is it room for
I'm sorry, what?
We give.
We need to get you more chocolate.
I mean, it probably.
Less chocolate.
Well, we'll see those at this time.
Much shallower. Yeah. We don't want to get to 700.
Just in case.
Oh.
Sure.
That'd be nice to try and get some if we can.
Okay. Thank you. I think that might be your job.
Well, I'll keep an eye. Kristen doesn't think we'll see them until we get to shallow.
Okay.
Next time come up.
Beautiful seafloor. Oh, small comb jelly. There's a lot of worms hiding. Going back uphill, aren't they? Mm-hmm. I think these were two large kind of bumps on the seafloor we were crossing before we reach a wall area, let's say. Or like slope. just have a quick stop and zoom on that large snail.
It's a snail with an emblem on the top.
Yeah, probably kind of completely covering the shell. Okay, thank you. Yeah, we can. Can we also just quickly zoom on the sea pen as well? Just since we're stopped. It's fine. Yeah. Thank you. We can continue to the point right now.
I think Christian wants the positive, this one. That is the other one. It's in the chat.
Okay, can we zoom in on again on this one quickly? We can back here for collection with the section sample. I think it's the same we want. I'd like to collect this for section sample 8. Can also be 6, 7, or 8 actually. here.
What?
Hate them. I hope it's still hanging around. There he is. I don't think he can run that fast. You could try. Yeah. Getting worse.
I mean, it's clear now.
Yeah.
Just to see it.
I can see it there.
Yeah.
Yeah, I know.
Patience.
So, that worked.
Oh, there he is.
Oh.
Yeah. Okay. Thank you.
That sounds.
There he is.
Thank you.
Hold on a second. Is there, you want to section sample some more stuff, or is that it?
Yeah, I'm actually looking. One of the targets was one of these fishes as well, but I'm not sure if you're gonna get there on time.
That?
Pretty sure Tom wanted one of these. If it's still possible.
Go for it.
Ah, no. Smart guy. Yeah, try. There you go. Okay, thank you.
That's in 7.
7.
7.
Thank you. It's all.
That was the thing.
It's a fish?
That was the fish.
Mm-hmm. Okay. Mm-hmm. Thank you. Now we go back to until waypoint 3, just a few meters more of transect. Can I try to find just a bit higher in this case? It's very dry slope, just a bit.
Sure.
Yeah. Yeah, that's fantastic. Thank you.
Okay.
Just until we reach a more flat area, let's say.
transects are away and waypoint for you.
Copy that.
I think once we reach about 1.4, we're gonna have kind of a higher, might have more stuff. Stop here so we can take one of the bags.
Sure.
Yep, one of the. And while we prepare for that, I would just like to do a couple of zooms here. No, just a zoom, just a zoom. Here as well on this one.
Okay.
And then we, yeah. Thank you. A lot of sediment again. That's what it is. And on the other one there was just on the side down there. Just to have a good look. Yeah, it's a tube anemone. Seems like it.
Is that what you want?
Mm-hmm. Thank you. And then yeah, I would like to take one of the saquitos, one of the little bags.
Which one do you want to do?
Did you learn the word saquitos, Olive? Well, the one on the well, they're both from the rock boxes.
Okay, let's do this one. One on the outside.
So the idea for the saquitos is more to take the first few centimeters, not go very deep.
Yeah.
Oh, nice.
Number one.
Mm-hmm.
No, great.
Any particular area or just?
No, actually, we just want to kind of cover, adjust the surface of the sediment so we can spread out along, and we can try to fill the bag a few times for that.
That good?
Yeah, it can be slightly more deep. Yeah, that's good. And you can just drag it around. Yeah, I guess. Yeah. There you go.
Change it.
And you can try to fill it a few more times.
That's your drag right there. It's like it's piling up, it's not really going anywhere.
Mm-hmm. It's very thick.
One more time.
Yes.
Oh, that's many times.
One more.
One more time. Where do you want to put this bag?
On the one basket. it's okay. Yeah, I know. But we wanted to keep the firebox a bit for later. So that's why in this case we are just keeping it outside.
Then do another for one more time.
One more, yeah. And then, yeah. You want to give more work to the guys? Sure. Okay. What can you do? Thank you.
No, I can empty it.
No. No. Just shake it a little bit in order to go in, and that's what it is. And we can put it on the raw plastic.
Okay, no plastic.
Mm-hmm.
Start with rock plastic. Must be one or two. Start with rock.
One.
Right. Outward. Unless you want it on the one on the porch, which one you want?
No, it can be the one inside.
Outward or inward?
The one where he was, yeah.
Okay. That's all the way out, right? That is all the way out.
Then inside the porch, really easy.
Now where'd you clear it up so I can see where you
Is this still helpful if I join? Sorry, it's still helpful if I join. I'll have to do
Oh, perfect. Thank you.
It's gonna stow that starboard, right? So
there anything else you wanted to do here?
If we can zoom in just here on the sea pen while we wait. Seems like it has a body. Mm-hmm. Umbellula. Beautiful. That's beautiful.
Can we hold it there like that for like a couple seconds?
So is that the time for CPAP?
Mm-hmm.
Not an optical?
Already. Thank you. Oh, so many.
It's a lot of sea.
Mm-hmm. It's quite beautiful. There's a lot of food around as well. Really a lot of them. Aaron, any ideas why we have so many?
Sometimes you get
A lot of
some
food?
deals, yeah. Conditions are right.
How are we doing on time?
Are we okay?
Transiting or we're...
No, we're looking for, right now we're on possibilities of sampling. We just need to figure out good sampling material.
Collections are possible?
Yeah. Do you see any of your target fishes?
Good one, but yes, that one.
There's quite a few. Can
There's a
we?
few couple of fish here.
Yeah. Can we have a look here? So sorry, guys. Can we have a look here on these fishes around? Yeah.
It's quite a selection.
Mm-hmm. can we park here? Try to park close to them and see if they don't run away.
See if it curls up when it moves.
So not a target.
Is this potentially a collection?
Potentially, but it's
Okay.
it's gonna be a fish, so let's see.
So if I step down, where
Tom,
there's actually.
that one?
I think that's Antarctica. I think those are the ones that we collected already. The ones
Mhm.
that curl up when they're upset.
Yeah. Okay.
There is a little ice fish right. at the
Right in
base of
front.
us,
Yeah. Just
but
here.
I'm not sure if we've overshot.
There's just one below.
So many of these guys.
It's just below there. Yeah, there's quite a lot of you.
He might be quite mobile. He might not be an easy catch.
Yeah.
Mate, where do
Yeah,
you go?
that
there we
one.
go. You know what I think? Looks like he's onto us.
Yeah.
It looks lively.
Is it one of your possible targets?
I think, yeah.
No.
We're not
It
gonna
won't
grab
be.
him. But
No.
thank you for giving us a look. That will let us idea it at least.
Okay. And there was another one just in front. We can have a closer look as well. Oh, it's a lot of Ketognathorome. Just, yeah, these ones.
I think one of these guys.
All of these are the same?
Are the Anasawacids, yeah.
Okay.
It's
Can we
weird how
just-
abundant
Can we just try to have a look at these ones? If possible.
I think these guys are all the same. They're doing
Mhm.
their donut trick.
That one is, yeah, even the small ones, right?
Yeah, I think that's a little crash.
Yeah. Okay, thank you.
Someone's helped me out in chat. Tremetonus scotti is probably the one that got away.
Okay.
But it's probably pretty quick for an army.
So quite a lot of pelagic, well, polychaetes around, and swimming polychaetes.
One colour. They colour them.
Mhm.
So that's
You're again a nice fish.
Oh, they're pretty quick.
Yeah.
Target. We think we can grab it, but I think it's going to be onto us.
Mm. Gonna try?
I'm getting that out.
Okay. The only thing is here is that every time we park, we have a lot of sediment in suspension. Do you think they run away with that?
With the,
With the sediment?
I think it might already be onto us. Being flighty.
Yeah, it's going away. Yeah.
Don't worry, guys. I
No,
think he's
no worries.
too aware of us.
Yeah.
Okay.
But thank you.
Okay. We would like to close the any skin bottle.
Okay.
Yeah. If possible, about a meter from the C4, or as close as the C4 as possible.
Here the station keep doesn't work as in the always like you keep station keep.
Now it's 2, right?
No, I'm asking.
No, 0.9 is fine.
Yeah, ready? Okay.
Mm-hmm.
Here comes number 2.
Thank you. Thank you. Okay. Still have quite a lot to have a look. I'm excited to see what we start getting along waypoint 4 and so on. Yeah, it's like good.
So anyone who's just joining us now, we headed down slowly for a while. Nice rocky outcrops with lots of filter feeders, coral reeds and things like that on the way down. We're now at the sandy bottom of this channel, and the transect across the bottom, and eventually will rise up on the other side. It's might get rocky and filter feeder again. So we're planning this dive, looking at these interesting habitat transitions, to get a feel for everything that's present in the area. And it's a big community shift. So at the base of this whole community is the material that's falling down from above. The surface waters are super productive. They're really green, and all that's raining down as this marine snow that we can see here, and that's what's feeding the community here. So you can actually see it if you look at the seabed. You can see the sort of more greeny, goldeny areas where there's material settling, and then all these animals are eating it, and that makes the sort of lighter areas where they've already cleaned up. So we see lots of animal burrows and feeding marks and poop, like that coil there on the front, left on the seabed, with the lovely German word Lebensspuren, meaning animal traces. So that's what's making all these patterns. It's the same sort of input, but as soon as there's a hard substrate, you see it flip from things that eat off the bottom, deposit feeders, you see it flip onto filter feeders. So things that get their filtering and feeding apparatus up into the water, with the exception, of course, being these sea pens, because they can anchor themselves in the seabed and then lift their feeding apparatus up. But as soon as we get to a rocky area, like this, even a tiny little dropstone is enough. You get things that are holding their filter feeding apparatus up.
Have any idea?
I don't.
No.
Blaine was flipping from urchin to...
Yeah,
I mean,
the
we could sample the rock.
We could.
Get everything.
Is it really rock under it? I'm starting to doubt even.
Oh.
Can I park here, please? No. This is some kind of bryozoan here.
You think the sort of these spicules are consolidating the sediment and you end up with this little mound?
Maybe.
Like a coral hummock.
Could be the sponge.
There's weird, like, tufts. Looks like a charity shop doll that's lived a full life and now it just has patches of hair. What's it in the Barbie film? Weird Barbie.
Has some ophiuroids on it, some bryozoans on the side.
Got a whole habitat in a handy little pick-upable morsel.
Any idea?
To me, it looks like a type of sponge, but
Yeah.
I don't know for sure.
But the spicules, so segregated, they're quite interesting. Sea mouse, Anna says.
Oh,
Gotta sample it, okay.
I think so.
Yeah, let's go for it. Okay, sampling this. Can we, well, on the bio box, on one of the bio box, can you try to pick it up with, yeah? Yeah, I think so. Yeah. You can put it on the Biobox 1A as well with a rock. Or sorry, on Biobox 4A is better even. I think it fits and then it will be separate.
I really don't know if this is even going to be hard or soft.
No idea.
I think
It
is
does
there a
has
rock as part of
below.
it?
Oh.
We were talking earlier about how the manipulators are slightly different. So we're a bit of a lobster. We've got a delicate claw and a strong claw. So this is the delicate one. We've got these gripper pads on it.
I'm not sure if it's a rock below it. Sometimes it seems there's something here, but
Locks into the claw and allows you to move it as one continual piece.
oh. Okay. So let's put it in Biobox 4A. So it's by itself.
4A.
Yeah. Oh, we do have a sponge expert on board on the chat. Yeah, it's proposing even a species already. It's also. Yeah, my box 4A.
Yeah.
Yeah. That's great.
Somebody in the chat was asking about how the sediment smells, which is a really good question actually. It's a sense that we're missing here in the videos. The ones we've been processing here actually like really rich, loamy soil. Like it's quite a nice smell. It feels still really productive and full of life. Things like the glass sponges, I've spoken about that a few times. They have a really unique smell. They are like a cleaning product almost. It's like a weird ammonia smell.
They smell kind of minty.
Oh, you got minty.
Yeah.
Ah.
Sorry, I'm right behind you.
No, minty's better. I've had some chemical smelling ones, but you never forget it.
But yeah, they smell mossy, like forest. I think I have a little bit in my office from the ROV, and I was actually showing Erin the other day. It was a little bit minty.
Huh.
It was very nice. It smelled like a rainstorm.
Ooh, that's a
Yeah.
great description.
I smell like a hike in my mossy woods to me.
Yeah,
Oh,
like
nice.
mossy woods.
Gonna continue. No, we can continue. Yeah. It's all good, thank you. We're gonna leave the other one alone.
We can try if you like. I think it's going to be quick.
Yeah.
The tail was already flexing.
It's gonna run.
Slurp might do it.
Yeah.
We get down the head end.
It's small enough. Yeah, that's true. one.
Yeah.
You're the fish man.
It'd be great to get it, but I think it's going to be quick.
I'll get away from this one. They won't get it. It's not strong enough.
Yeah, that's true.
Not much room.
What is it? The seats.
It's here.
Seat up. Is it? I'll prepare.
There's something already in 6.
No, it's not in 6.
There's something in 6.
It shouldn't be. Oh, it's a fish.
Oh, it swam in itself.
Oh.
Go, catch your fish.
Okay.
Actually, the ROV has
It should be
accidentally
on-
been catching those. I think I'm up to about 5 of them.
Well, it should be a fish on 7.
Yeah.
Okay.
There's one in 6 too.
Sure.
Can have company.
Okay.
Okay, is that? Okay.
What do you need?
You sit up, get the tilt up on the seat. All right.
Sorry.
Okay, it's okay already.
It's sucking away. Further out. Technique.
Sneaky, sneaky.
Oh, dude.
Too lively.
Ouch.
Thanks for the attempt, though.
Thank you.
It's,
It's a fish, you know. You can swim.
Hold on a second. I don't see the fish in 6 either. Swim out.
Thought I'd come from behind.
No.
I think the fish got out of 6 too, Joel.
Uh-oh.
See, you lost him.
That's okay.
Oh, wait,
Two
no, there
in
he is.
one.
There right now. He's
The-
still there.
I know he's there.
There'll be about 5 or 6 of them stuck in the frame somewhere. They keep turning up after that. Yeah, they're idiots.
They are. You also just heat the seafloor. Keep on heating the
Yeah.
seafloor and just get stung.
So these really iridescent, sort of silvery fish that we've
Do
had.
you want to keep that one, or do you not care about him?
Don't care about him so much. If you want to eject him.
Oh, we
I
can,
wish I could.
we can keep it for now.
Yeah.
I'll just leave it on 6. If he gets
Yeah.
out, he gets out.
No worries. No worries. So yeah,
Thank you.
these silvery ones are,
Let's continue.
I think at least Notolepus coatsy. And I think it's the Antarctic Jonah fish, I think it is. They've been hanging around the ROV a lot. They seem to have no fear at all. And they are, I think I'm up to 5 or 6 of them that have just been captured by the ROV's frame. They get themselves wedged into the tool tray, they get themselves wedged into the frame of the ROV. Yeah, after every dive so far, someone has found me a fish wedged in somewhere. So they're sampling themselves, it's pretty handy.
So we can continue.
These little donut guys that are going past are a eel pout.
Beautiful jelly.
And this is their defense mechanism, is to coil up like that, which obviously works against their regular predators, but does not work very well against an ROV. So they're like Adaptus antarcticus.
We continue to on the direction of
There's
the
2
transport.
species actually, and you can just tell them apart by
And you can see something
the
really
relative
nice.
proportions of the head.
One of the
But the ones
priority
I've caught
groups.
so far.
We're looking also for mollusks, some raccoons, some corals. Missing what am I missing? And some polychaetes as well.
Red RV, you have another jet, red one.
Update away.
Thank you. did take it off.
Manual. Right next to it says manual. There you go, thank you. No, it's good. Just go ahead and leave it on it.
Put it out, so.
Yeah.
So what do these guys eat?
Take it out. Which will stop doing.
Your little curly
Coelicoids, invertebrates.
invertebrates. So that's
mainly.
why they're always going crazy to the seafloor every time they see it.
Oh, the ones that are crashing?
Yeah.
I think they're almost exclusively copepods.
Ah, okay.
So I think they are. I don't think they're hunting when they crash into the seabed. I think
Okay.
they're startled by the ROV and becoming disorientated.
And these ones are just going for the seafloor and search for whatever little creatures they find on the sediment.
Yeah, all these oacids are going to be probably invertebrate predators.
Are they selective at some point, or is it known?
It's tricky when you're in darkness.
Okay.
So they're probably just snapping at movement, although there's pretty good eyes on them, so they're probably looking for some bioluminescence as well. There's just so many of them. And there weren't at the other sites. They were quite rare at the other sites. We get one or two.
Yeah, and here they're everywhere. Way more sponges than we have seen until now as well.
Because this is so common, I think maybe we should get a small one that size. Yeah, I think it's common here. We should probably get it to your presentation.
Okay.
And I'll just give it to Christian and he can see a sample for me for
Okay.
DNA.
Sounds good.
Just it's
So we can see it.
incredibly abundant.
That one?
Yeah.
In a coral?
In a coral quiver, yes. Do you want the whole
I
thing?
think if they can, just grab the whole thing, just plug it.
It doesn't fit the whole thing on the coral quiver, I guess.
It might.
With the
How
stalk?
long is that? How long is the coral quiver?
With the laser. With the laser.
I think it'll fit.
You think so?
It'll fit.
It's a few amphipods, I think, on the stalk as well.
Yeah.
That's interesting.
This one's pretty short.
Okay.
If the stem sticks out, it's okay.
Yeah. Okay. Hey, man. How are you?
Yes, all right.
Okay.
You want eight?
Just a tourist.
Vamos shot.
Yes, please. It's just like a flower.
It's beautiful. Can we also have a quick look as well in the stalk? Because I think there was something attached, but I'm not sure. Oh wow, that's beautiful. Yeah, maybe not. Yeah. Thank you. That's a beauty.
It's got nice light. Oh my god, you put this backwards just so that I.
Here's something for Alex.
My brain. Hold on.
My brain.
Thank you. There we go.
That's incredible.
I mean, that is ever.
Ah, there's some beautiful.
Taxonomist dream videos.
What?
It's like the dream video for a taxonomist to have all this detail.
Oh, yeah.
Mm-hmm.
Oh, there's a little guy.
Mm-hmm. There is. A little.
That's fine.
Soon
Could
for the
also be
fabric of
partial.
reality.
It's very difficult to distinguish them.
Yeah.
Oh, it really struggles if I zoom in that
Yeah.
much. That's weird.
Because they're moving.
I just want to see that little guy. Sorry. Alright. You guys continue. Thank you.
Alright, so.
Okay. To
This
the.
one's going?
Let's do Vancouver, yeah.
Yeah.
So, yeah, 1 or 2. Okay.
This works, right? This escapes predators.
Sure. If you think so.
We were chatting earlier about the ROV being a new apex predator, because that escape response obviously has worked for millions of years, but
Definitely helps you.
It's in my ready, yeah.
not screaming.
Sorry, we're
Why are you
making
screaming?
an eye conversation.
You sono you don't.
No, it's good.
Well.
Yeah.
I'm sorry, you can put it on. I think that.
That's all right. Thank you. Okay.
Thank you. What?
Stuff from Montenegro.
That's the name of the sediment. Yes. Yes.
Beautiful green art. I like those.
So in between our sampling stations, we're transecting over the seabed, and you may have noticed those two little green dots right in the center of the frame. So that's our laser scale bar. That's 10 centimeters across, which will give you a sense for how big some of these animals are. But when we are all back home processing this video, that will let us make quantitative assessments of how dense these animals are.
And even measuring their size,
Yeah.
like biomass wise.
Oh yeah, actually converting it to mass.
So that's why I'm always asking lasers. They're super useful.
Yeah, but super easy to forget. It's just
Yeah,
a couple of
it's
little
true.
green pixels.
That's true. Totally understandable. Wow. Jellyfish world.
Oh, a little trio.
Yeah.
Setting each other
way
away.
more. Wow. There are quite a few actually at the bottom as well.
It's a great shot. How are they
Wow.
ganging up in just one spot?
There's one at the bottom there. Here. Oh, quite a few at the bottom. Just have to keep a close eye, but oh, that one was hiding. That was an aquarium. Just, yeah, it's everywhere. And more. Oh. Wow, there's dozens of them.
I hear Alex.
They're everywhere.
There's a swarm of jellies. This is great. Why all in one spot?
Wow. Very interesting.
There's a good question in the chat about what's the weirdest fish behavior. There is a lot to choose from. I do have a... Oh, there's a nice fish here. That's a plunderfish up close and then a nototheno further back. Yeah, I like spoiling people's day with kissing gouramis. That's a good one. People like to get a couple of those and just like, they're kissing. No, they're fighting. That's how they could compete with each other. So kissing gouramis are actually fighting. With all these sea cucumbers as well, I think one of the highlights is going to be the pearlfish, which live inside cucumbers. They don't seem particularly happy about it either. And if the front is taken, then one of them lives in the back. So they spend most of their lives in a sea cucumber's bum. It's apparently a safe place to live. The fish that we're seeing here in quite high abundance. So there's these eel pouts, the ones that are curling up as a little donut. They tend to be closer to the bottom. They're like adapters, we think. And then there's long, thin, silvery ones that have been shimmering along. I've been debating that a little bit, but as the chat got right, we're leaning towards Notolepis for those ones based on where the pelvic fins are. And those are the ones that have been capturing themselves in the ROV. I don't think we've deliberately sampled one, but I've ended up with 5 just because they keep getting stuck in the tool tray. And we currently have one of them in the slope gun rosette that captured itself.
Come up to a point 4, please.
Big tube line. Is that some of the burrows we've seen just sticking out, but that's one fully excavated?
Seems like it. There's also a lot of darkness, and you see some of the burrows just being points. You kind of see like
Oh, a little
some
puff.
sediment goes out. Yeah.
These little skinny guys sitting on the seabed, I think they're Bathydraco. If we got a chance to try for one of those or to get a look at, they've got quite flattened heads and they're sitting on the bottom like a crocodile.
I'll have a look. Like the one in front? No, the one in front looks like a rockfish, no?
Yeah, that's another ice fish. They've been wise to us. There's been some
Not
coming
this
past
one.
that are a little bit paler.
Not this one, right?
No,
Oh.
no, they're a little bit paler. The eyes are right on top of the head,
Yeah.
and they're sitting quite low down.
It's like this silverish body as well, like we said last time.
It's got some pattern to it, but it's lighter than the other ones. It's kind of a sandy yellow,
Mm-hmm.
and then with the eyes right on top.
Like this one's?
Okay, we'll keep transiting. I'll try and shout out when I see one.
Just keep an eye on it
Yeah.
and let me know when you see it.
What's that on the right?
Think it's that? You have it in there.
I think that's a plunderfish.
That's the one we
maybe.
missed. We tried to sample, right?
They might be a bit quick, but yes, that's with the black spot. Yeah, that'll be onto us, but it's a nice shot. We'll be able to ID from that.
Mostly a shot, right?
Yeah, that's fine.
I hope, well, I think things will start change soon. Based on the topography already. Wow, that's a huge anemone. This place is beautiful.
Yeah.
Full of life.
We may see a transition coming up as we're up to our next waypoint, or soon. We're going to start to rise up the other side of this little trench. So things might get rocky and interesting again. Not that it's not been interesting, but we'll have a habitat transition.
Oh, small tunicate.
What's it sitting on? Is that a rock?
It's a small rock.
How are we on our rock collecting?
Well, we should have one on the next one. Well, around this waypoint. So let's see how if Andres sees any interesting rocks around this waypoint.
That's it.
But no, not yet. Interesting.
Is that a crinoid spread out?
Mm-hmm. It is.
So where's our laser? That's like 20 centimeters across.
It's huge. And the wall, were you here when you saw the wall full of crinoids?
Yeah, I was watching from the base.
What's that fish doing?
like how I have to explain the fish.
Yeah, sorry.
What's that one doing? Why is it like that?
Half of the time they don't know.
Well, this is cool because a lot of these species we don't have any video of.
Mm-hmm.
It's good to see how they live. It's good to see which ones make donuts.
And you can see, so that is known for like a stress response.
Yes.
Let's say, like right now.
Yeah,
Okay.
there are pelagic varieties as well that do it too. And there is a few. Every now and then there is one that is different, but it's hard to.
Yeah, like that one on the left. I don't know.
It's hard to stop an undersea bus immediately. An octopus on the right,
Mm-hmm. That's all right. Can we so
sporting a different pattern.
quick look here in the corner? It's still possible. No. Yeah. Just there.
Nice pajama stripes. And there's a fish being a donut. Oh, two.
Oh. Mhm. And this time does not seem to be a Dumbo.
The curled arms.
Yeah. So cute.
Bye.
Oh. It's really small.
It is tiny.
Perspective-wise? No. No, no, no. Don't. Oh.
Little isopod?
Oh, yes.
Now, were they on the hit list?
They are, but we do have these. And this is not one of the target ones
Oh, okay.
anymore. Now we're looking for the trilobite one.
Right.
Right. Yes. Some tunicates. Some different tunicates coming up. This one's you see them, yeah, you see them kind of up on the sediment. Pretty cool. Oh, can we stop here? Just to have a look at this guy.
Oh, nice.
Here, and then if we can there as well. Can we get a good zoom in? Mm-hmm.
Very
We got
cool.
one yesterday.
Yeah, he raised up that.
Yeah, I saw that yesterday, yeah.
Do you still call them sea mice?
Sorry?
They still call them sea mice? Is it sea
We
mouse?
did.
It's where the common names vary so much geographically.
Yeah, that's true. And just have a look what this is here in the corner. I think it's maybe a mushroom pool or just a sea bed. Yeah. Leftover. Yeah. Yeah, you're right. Mm-hmm. Okay, thank you. We can continue.
Do they want the wine?
No, we already have this
Oh,
one.
good.
We can continue.
Someone was asking if we ever used bait to capture some animals, and that's absolutely my favourite thing. Some of these mobile fish may be the only way we can catch them. So large mobile scavengers and predators, baited traps and baited landers are the way to go.
Not a happy fish.
Already onto us. It's not to be annoyed.
I do like when they look cranky and unhappy.
It's something fish do really well.
Yeah.
They can look furious.
Sorry, can you zoom in as well? Right on there. This sponge looking. It might be the same, but just to double check.
Which ones?
This one just here at the bottom. This one's here. Just for a quick look. Yeah, just to confirm, yeah. Thank you.
Bridge, RV, updated waypoint.
Updated waypoint.
So if our mapping is correct, we should be coming to the end of this sandy area and then start to climb up the other side of the ridge. So things should get rocky. We should get more filter feeders and some good views as we climb up.
We can stop here. I want this one. Rock. Let's confirm, but I think so. Yeah. Other? Please. Yes, and it goes to Biobox 1A. Yes, 1A. Then do not worry. And you continue. Yeah. Next one, right? Yeah.
Bridge, RV.
Yes, RV, go ahead.
Yeah, are you on the move or no?
Negative, sorry, on the move now.
Yeah, right.
We changed the language from English to Spanish right now.
Hello, everyone.
todos y bienvenidos a este buceo en el mar de Bellinghausen. Mi nombre es Patricia Esquete y soy la jefa científica de esta expedición hasta la parte más interna del mar de Bellinghausen. Estamos en este momento buceando a 885 metros de profundidad, justo enfrente de una banquisa de hielo. Hay que decir que esta es una de las partes más inaccesibles del mar de Bellinghausen, porque normalmente resulta muy difícil llegar por causa de tanto del hielo marino, que es el que se forma encima en la parte más superficial, por las bajas temperaturas que existen en la zona, como por la gran cantidad de iceberg y el desprendimiento de hielo de esa banquisa. Estamos andando por un fondo sedimentario, es decir, por un fondo de arena. Fango. Y vamos a ver qué nos encontramos, qué sorpresa nos depara. Wait, we're gonna sample the rock. Yes, please.
Yes, RV, go ahead.
Hey, Jason, we're gonna pick up the rock here.
Have fun. I'll bring the ship this up.
Thanks.
Where are we going to put this position?
We're gonna put it in Biobox 1A.
1D?
1A.
1A.
Hola, hola. Complementando lo que decía mi compañera Patricia, bueno, yo soy Cristian Aldea de la Universidad de Magallanes y estamos aquí justo enfrente de una banquisa de hielo o plataforma de hielo, en inglés ice shelf, ¿cierto? Que se denomina Bach, justo en la península Beethoven de la isla Alexander I, en pleno mar de Bellinghausen. Seguimos con estos inmersiones, buceos de profundidad. A este momento, como decía Patricia, estamos a 884 metros, justo muestreando una roca, como ustedes pueden ver, que sobre su superficie tiene una hermosa fibra, ¿cierto?, del grupo de los equinodermos, y en este momento el brazo se encuentra capturando o tomando, haciendo esa roca, ¿ya?, para depositarla en una de las cajas dispuestas para ello.
You can zoom in to the rock a little bit. Esta roca bien interesante, tiene mucha vida, además de los fiura.
Correcto.
Hay un isópodo que está a punto de caerse y
Uy, ha caído el isópodo.
perdimos el isópodo.
Se ha
Existen
salvado.
braquiópodos,
correcto.
un erizo,
Como una esponja arriba.
Una esponja arriba.
Sí.
Yeah, that's beautiful, thank you.
Y algunos, no sé si poliquetos tubícolas se observan también.
You said in Biobox 4A, correct?
1A.
1A, okay.
Yeah.
Lo importante es destacar que esta península Beethoven prácticamente nunca ha sido muestreada en lo que es el área bentónica, porque justo estamos en el borde de la plataforma de hielo Bach, como decía, que es una de las plataformas de hielo de la península Antártica Occidental, ya justo al lado de la península de la plataforma de hielo George VI, que también es bastante grande.
1A,
Ruby Bridge.
Go ahead, Bridge.
Hey Ben, would it be alright if I brought the ship setting to 270?
That sounds wonderful.
Wonderful.
and also before I take off.
If we can zoom in a little bit to the sky and that one, just to kind of be able to know who they are.
That is
Who they might be. It's a beautiful thing. Now we have a glamour shot. That's good, thank you.
You happy?
Very happy.
Okay. You want to do the other one on top?
Just a bit. Yeah, that should be fine. Yeah, just like that. We don't need more than that. Thank you. Seems to have some stuff on top. Some polychaetes. Yeah. Thank you. I think we can keep going.
Harvey Bridge.
We are continuing our transect waypoint 5.
Excellent. On the move.
Podemos observar allá una concha de molusco, pero evidentemente está muerto porque está hacia arriba. Y un poliqueto nadador. Erizos irregulares, que son esos rojos que se observan en el borde de la pantalla. Y varios ejemplares de umbélulas, que son esos nidarios pedunculados que se observan en la imagen. Nidario del grupo de los penatuláceos umbélulas, cipens. La pequeña medusa ha pasado rápidamente nadando.
Sí, a la medusa le gusta pasar por la cámara.
Justamente se observa también esos poliquetos que tienen una natación bien especial, en particular como forma de S zigzagueante. Esas anémonas gigantes también. Anémonas más pequeñas, anémonas de varias especies. Un erizo regular al lado de una anémona.
Seguimos encontrando muchos de esos pececitos.
Esos peces como vermiformes tipo zoárcidos son bastante comunes en estos ambientes. Un gastrópodo grande que probablemente la especie Arpovoluta charcoti cubierta con la holoturia, con la anémona, ¿ya? Justo estaba en el pie del hombre. If you see scallops, yeah,
That's what we want. Okay. If we see scallops, then we want them. Just a heads up.
alive.
Yeah, I like.
Yeah.
But only we find them. Meanwhile, we can keep going.
Esponjas hexactinélidas por allá y una hermosa anémona. Cabe destacar que el grupo de las anémonas no es tan fácil identificarla a simple vista, así de rápido. Sin embargo, con las capturas nosotros podemos hacerlo o al menos inferir una especie. Eso que se observa abajo probablemente sea un briozoo, una colonia de briozoo. Tiene todo el aspecto de briozoo. ¿Cómo está salido de foco?
No, esto pensamos que era algún tipo de basura, pero parece que es una roca.
Es una roca. Una roca.
En primer momento estamos sorprendidos de encontrar basura aquí, pero de hecho no, es una roca. It's a rock. We can keep going, thank you. We're expecting to find something more interesting stuff around the next waypoint.
Se observa el comportamiento clásico de los erizos que se cubren con restos de conchas de molusco y otros animales se posan más encima.
Can you zoom into the rock, please? Just to see who's living in that rock. So it's good to have an overview of what is in there. And here.
Sobre las rocas se observan algunos braquiópodos, ófiuros. Cuando el vehículo se intenta pasar sobre el fondo, evidentemente, debido a los sedimentarios, el carácter sedimentario del fondo, se resuspende mucho sedimento, como pueden ver.
Como la que recolectamos antes. Esponja misteriosa.
Esponja misteriosa.
Y nos costó un poquito averiguar qué era. No tiene la forma ni el color usual de una esponja.
Estrella, una ejemplar de estrella. se observan muchos depósitos de concha en el fondo marino.
Sí, te lo iba a comentar en este momento,
Sí.
que parece que el fondo ha cambiado y tiene como grandes depósitos de concha.
Sí.
Pero no sé hasta qué punto. Ah, esa estaba viva, ¿lo viste?
Sí,
Gracias?
sí, sí, sí.
En realidad son animales vivos.
Algunos sí. Y hay braquiópodos también directamente sobre el fondo marino. Sí. Ahí hay ostiones también, scallops.
¿Dónde están?
Probablemente los que están debajo.
¿Me lo señalan?
Esos anaranjados, esos rojos que están abajo.
Ah,
Sí, Santi.
no lo vi. ¿Está aquí?
Sí, tal cual.
Can we try to
Zoom
sample that
first,
one?
please.
Zoom in,
No,
zoom
close
in.
up.
Yeah, you're right. It was totally a scallop that
Yes,
we'll never,
scallop.
we'll never see again.
That was quick. We're gonna pick this one up.
I have the hope that here the sediment is not that fluffy. Maybe
Bridge, already.
Rugby Bridge.
We're gonna pause here for a moment.
All right. I'll stop.
do you think would be better to suction or
Pick up? We'll see.
everybody trust you?
Where do you want to put this? If we're going to put it somewhere?
I mean, if it's, I think it can almost go everywhere. It can be a firebox, or it can be a suction something. Everything will be fine.
See that big thing? Yeah, I think if it was me, I would pick that up with a small soft jaws. No, soft jaws. It's up to you. You want to suction it? That's not going to swim away.
That constantly got good.
Yeah. Location. The whole limit.
Ahora esperamos un poquito a que se redeposite el sedimento, nos dé más visibilidad. Y los pilotos, entre tanto, están están comentando, están intentando hacer una estrategia de cómo muestrear,
Sin posar el.
cómo muestrear estos bivalvos sin romperlos. Una posibilidad es con el brazo y mucha sensibilidad. Otra posibilidad sería con el succionador, que es como una aspiradora. Pero luego se puede romper siempre también al llegar al contenedor. Y tenemos justo uno en medio de la cámara. Hay varios, de hecho. Este también es
Sí.
un scallop, ¿cierto? Hay uno,
Creo que
dos,
sí,
tres.
tres.
One,
Bien.
two, three. Any of those?
I think because you want all three.
If possible. Otherwise.
It's faster, yeah.
Ah.
But if you think it's safer to just grab it, do what is safer. We want
Suction.
them? Yeah, perfect.
Yeah,
We
that's
want them,
fine.
but we want them whole.
Yes, there's three.
We completely trust you. No, thank you. Un poquito menos fino, sí siendo fino, pero un poquito menos fino, sí siendo fino, pero un poquito menos fino. Y empezamos a encontrar otras especies. It can be a big one. It's 25.
Jaffa.
I'm gonna just turn it on.
not sure if it's alive. Looks like more broken. But, yeah, that one looks broken though.
Yeah, maybe it's not alive. The
Yeah,
last,
we probably
the
don't
last
need
two.
this one.
You don't need this one?
No, let's try to zoom out.
What about that little guy up there?
I'm wondering, yeah, maybe it's alive. We can try.
Which one?
This one?
Yeah, okay, let me zoom in
Oh,
and see if
this
that's
one
a
maybe. This one looks alive.
Yeah, let me see that one.
Oh, that's adaptable.
Is this very small
Yeah.
or something too?
Yeah, we can try.
Okay, let me zoom in.
It doesn't hurt to take it.
That one?
Yeah.
Okay.
Yeah.
Okay, I think this one on the left, that might be.
White one?
Let
No, I think
me-
it's dead, isn't it? That white one's dead.
No, I mean, I was thinking about this one, but there was another one more to the left.
Do you want that one?
Yeah.
Who wants that one too? Okay. That one? No.
Yeah, this one. Sorry, I was pointing wrong.
Okay.
Yeah, that's it. Really alive. And then a little bit more to the left. I thought I saw one. This one here.
That one. Is that right?
I don't know. It looked better than in the close-up.
There you go. Let's see, it's a stretch.
Mm.
Yeah, you got it. Stretch. Clear, you can see. Clear the push course. Okay, suction's on. There you go. Oh, that looked like it was alive.
Yeah.
Yeah.
Is it helpful?
Is there another one?
I'm not sure. No, it doesn't look like the same. Maybe this one. Here.
You see it?
Yeah. This one looks more
It's
like
a brachiopod.
That's a brachiopod, yeah.
No?
It's a
Yeah,
brachiopod.
It's not gonna come because it will be attached. Or it will
No.
take a while.
Looks
We
like
don't-
it's gonna come.
Yeah.
Try and flip it.
No, we don't really need
Oh,
it.
you don't want it?
No, it's not the one we need.
Is
Thank
there
you.
anything else?
Anything else, Christian? No, we're fine. Thank you very much.
Alright. I'm gonna put this in full.
There are quite a few.
Oh, there you go.
It's confusing because they all have the same color.
Sorry. Good. Let's watch. Right outside. I'm not a fan. You're not? Yeah, hanging out here. I got such knots. Okay. There they are. I was wondering where they were hiding. They all shoved to the top. And then index to 4. I'm in 4 now. Okay. That's the arm. Lock and Stay 1 at 5. Okay. Shall we?
Okay, yeah, we can continue. Now we're probably coming up. We're coming up slowly. A little bit of slope.
We are slowly coming up. Right now we're at 7.
se puede observar un hermoso ejemplar de Sidaris, Sidaridus.
It's really, really steep, and yet full of sediment. To be honest, this part I thought it would be a little bit more rocky. You never know. Completely wrong. It's a promising rock.
Una pared de rocas magnífica.
It's a wall. See that as well? Get arranged out so far.
Can we do something with the color? I'm sorry about that. It does. It's kind of like. Is it the line? Or?
Something about the kindness. Yeah. It's gonna be easy, there's so much water. Back up a little bit. Yeah. I think it's just if we want to get a better view, we have to get closer.
Darby Bridge.
Hey, Luke, I think we might take a look at this wall for a minute.
Copy that. And just to let you know, we do have an iceberg ahead of us that's headed in our direction, so we'll have to come a few hundred meters to starboard in the not too distant future.
Roger that. Do you have a time estimate on that?
Bueno, y ahora
15 minutes, we'll probably have to start moving.
okay.
Batty track, maybe.
You got it, Luke. And another question. Do you have an approximate speed you'll want to make up through that?
Maybe 0.4, 0.5 would be good.
Okay, we shouldn't struggle too hard with that. That sounds alright.
Should be doable.
Basically race car down here, Luke. You know that.
Why is it that we can? It's because it's all the material.
Silty is coming from top.
And that's why it's pretty much is greenish. Kind of difficult, huh?
I've got a big overhang on top of me.
Bueno, pues acabamos de llegar a un hábitat completamente diferente. Tenemos una pared de roca delante de nosotros, muy interesante. Pero toda la materia en suspensión está haciendo muy difícil la visibilidad. Entonces vamos a esperar un poco a
Can you move forward? That looks like we have about 8 meters.
Vamos a ver cómo nos podemos mover para conseguir una buena visión de esta pared. Al mismo tiempo, hay un pequeño contratiempo que ya no es la primera vez que ocurre durante esta misión o durante esta campaña, y es que se acerca un iceberg y tenemos que movernos un poquito hacia estribor, mismo que no estuviera dentro de nuestros planes, para esquivar ese iceberg, dejarlo pasar. Aunque a pesar de que esta pared es tan interesante, va a ser muy difícil que podamos quedarnos a muestrear o a contemplarla.
Which way did you say that? We're going to move a couple hundred meters to the east. I can't tell if you're going to well, we're definitely not on top of it yet. You have to look at Patricia.
Sí.
Because in about 15 minutes I can't stay.
Sorry again?
In about 15 minutes I won't be able to stay.
Yeah, no
so
problem.
See what you want to see. You want to keep climbing
Understood.
up.
Can we close an skin bottle before?
Can we get on top of this?
Yeah, can be.
That's pretty,
Yeah. Can we? Will we be able to sample it?
Which
Sample
one?
the
Which one do
coral?
you want?
Can you point it out?
Yeah, here.
Yeah. Yeah.
This one.
Justo se está solicitando el cerrado de una botella Niski para tomar muestras de agua en este momento. Así que el vehículo va a proceder a tomar una muestra con la botella.
Start with rain.
We can be.
ROV.
Hey, Luke, we're going to try to get a coral before we have to leave, but on your go, we will drop it and start moving.
Ask him how much
Cool,
time we
yeah,
have.
you guys have time for sure. 15 minutes was quite conservative as well.
All right, appreciate that.
So if we can start.
This one.
It's very complicated, but we can start with the skin as soon as we are stable.
Roger that.
Yeah. Fantastic. Now we have a beautiful and nice view. There's quite a lot of life in here.
¿Batitraco? No.
Perhaps this fish.
Send
So if we
markers.
can start with the skin as soon as possible, then we'll try, well, the fish left, but yeah.
Okay.
Alright, so this one's up there. Set the file down to 27.
Uh,
Okay, we're
yeah,
going to do this
we're gonna
first.
start with the skin because that's something we need either way.
So this will be 3?
Yep.
Can you increase the, there you go. Thank you.
Oh, I gotcha.
Skin. Extending.
Roger.
Man, team.
Boom.
Number 3.
And then T. You want to do
And then I zoom in to this. We wanna check if it's a coral or assess what it is, maybe bryozoans or polychaetes even.
some more?
I think they are bryozoans. They are bryozoans, so we don't need it, but it's great that we got this beautiful view. Thank you.
And polychaetes.
Are you happy with that?
Yeah.
Acidians too.
So let's then continue exploring this wall.
Okay, no sample.
I guess not.
No.
Which one do you want to look at next? Is there anything you want to look at?
Oh. I'm gonna zoom in.
Correct.
See if I... Yeah, we're gonna connect this one. Hopefully it comes with the isopod, which is that little
You want to
guy
take
on
it
top.
first?
Yeah, a little bit. You can have a nice view of it.
Take that from me too. I think you can.
Yeah, it's right in the middle. You want the whole thing or just a piece?
Well, hopefully just a piece, but it's quite small, right?
Yeah, it's quite small.
How small it is?
I think we just have to pull
Yeah,
it out.
just take it. Let's just take it.
Al hacerse se puede observar también la presencia de numerosos briozoos sobre la roca,
And then the next
directamente
zoom in
tapizando la
one
roca.
you have in the hand.
Aparte del coral que se veía.
Coral Crater.
Ahora se va a muestrear ese coral y se va a depositar en contenedores diseñados especialmente para los corales.
Let me give you a better picture. How's that? The Bach. And you need to rotate your jaw.
Yeah, I'm gonna cut it or pinch it.
Are in the same place of the
Is that bad?
Yeah.
Blade at the back. Yes.
It's gotta be cut.
Yeah, I think so.
Will this pinch off, Patricia? Or do you think it has to be cut?
Sorry?
Will this pinch off? Is it hard?
No, it's probably squishy.
Squish. Since it's so small, I'm not sure the fingers, just the soft.
Okay.
Squishy so you can get close as possible to the base.
Oh, I see the slip.
It's a picture. You got this. It's beautiful. This name carries that blood. Beautiful. You got it. Or no. Pastor.
Better than the best, man.
And she wants a burger shot.
Yes, please.
You want to give it a 90-degree turn?
That would be fantastic.
Mhm.
That's a beautiful one. We need the polyps, but no.
You wanted the glamour shot.
Well, you cannot have everything, I guess.
Right?
I feel particularly attached to those kind of animals, but you cannot have everything, indeed.
happy?
Happy, happy.
Okay.
Similar.
Someone might come
It's an autotonic, but I don't know what species. Maybe this Osticus.
Perfect. Champ.
Get in there.
Fantastic. Yeah, it's gonna go.
Ow. Nice.
Not with my isopod, but still in there, intact.
In the bob and her.
Okay.
There's one more thing.
Stand by.
Yeah, that was a great job. We want this white tubes and one of these transparent things.
Quiver? Coral quiver?
No, it can go in the firebox.
For the white tubes?
Yeah.
Okay. It looks small. You just want to mix it in.
Yeah.
That doesn't really matter as long as we can find them.
Okay.
So one of the small biobots can be 4B or 4C.
Not empty?
All the white tubes.
White tubes. And that big bubbly thing, I think, is what she wants.
Yes, that thing. We can zoom in them first.
Hold on.
That looks fine.
Maybe star thing.
Well, beautiful.
It's a tube worm, isn't it?
Yeah, these are two worms and there are some hydrozoans.
So it's just gonna grab that whole clump.
Yeah, I mean, a few of them, at least, few grabs. It's probably some crustaceans. Having fun in the middle. And then yeah, the bubble thing, I'm not sure what would be the best way to get it.
I'll leave that to the master.
Ah, totally.
Master Tyler. We can hinge that back a little. That's out there.
Como se puede observar, en una operación de bastante precisión la que se hace para colectar ese tubo de poliqueto, tubícola. Evidentemente va a salir con trozos de hidrosol. Es muy complicado. Pero yo pienso que se va a poder hacer.
So the tubes are probably fragile because they are carbonate.
Circular, tipo circular.
But if we take them from the base, that would be fine.
Is the base on top or is it the base
So that where you had it, that's the base.
Yeah.
Yeah. That's the base.
Perfecto.
And the Bach. Glamour shot.
Perfecto.
No, we don't need it.
No glamour shots. Roger that. Okay, where do you want to put this?
By box 4B.
Number?
4B. Bio box 4B.
4B.
The bubble.
The bubble.
Yeah, well, the bubble might be squishy but delicate. I don't really know how to. I don't-
Bridge.
Go ahead, Luke.
Yeah, just to keep you updated, that iceberg's slowly moving to the north now as well, so there's no rush. If we just stay here, it should actually pass us.
You are the man. Thank you.
Are you saying that to Phil or to the iceberg?
No.
To Luke, sorry.
Not pressured. Okay. Well, we can try pinching it.
Yeah.
And then, if that doesn't work, there's several. We can suction it slowly.
Mm-hmm.
So
Yeah.
we'll try pinching it first.
Yeah, let's try pinching. Let's get a close up first.
I'll do this one.
Because,
What do you think of that one? This one?
I think that might be the easiest one with you.
The middle?
Yeah.
possibly this one you can take it from the base.
Yeah, that would be
Yeah.
the easiest.
It's the best.
Something. Agreed? You're all in agreement? Better picture. Oh, sorry. I was doing the other one.
Looks good.
It's the brain.
Yeah. Like it has veins or something.
Se puede observar directamente los sifones de la... Sí,
Yeah,
directamente.
fine. Thank you.
Okay.
Yeah.
Okay. Show.
All right, let's see how
What do we do on this one?
Yeah, a bit of a
Where is this going, Patricia?
This can go to the same one.
Actually, before?
Before, yeah.
this weekend thing. Yeah, just trying to like kind of peel it off. Yeah. Close my jaw so a little bit more so you can feel it. Yeah, so you catch it out. Yeah, my decades of experience.
It doesn't come complete and suffer too much. Mm.
It's start wrapping up. Like, kind of like, kind of grab by the scruff and all that kind of stuff. Oh, no, not there. In the middle.
good one. It's quite amazing.
Excelente.
We're gonna B4, stand by. Sorry, buddy. Okay, B4 coming out. Okay.
B for B is fine.
B4B.
Before boy.
There you go, you're in, watch it. Go. Call it back here. Go in the Bach. Oh, no, it's still there.
Yeah.
It's stuck on just. Give me a little bit of zoom on the side. Roger. Yeah, good sit video too. It's a picture. Okay. I just have to open my jaws a little bit more. Yeah. There you go. She's dropping. Get down there. She's down.
No, it's still stuck.
Yep, that's it. Okay. There it goes.
Excellent.
Wonderful.
Okay. Watch
Okay.
it. As you can do this.
Yeah.
It's doing well.
Perfect. We have more requests for here. So we want to section this sediment
Okay.
to number 1.
Shot number 1. There. When you get to surface in that thing, it's gonna hurt. I think.
What?
That one. When you go to the surface. We're on stove. Okay, which one?
So just this sediment here.
So
To section sample 1.
I think we should be able to reach that one. Which one? The top of this one?
Yeah. Yeah, we can always ram it into
Roger. Okay.
Shrimp.
We don't really need a trim, but if it comes with a sediment, so be it. We don't need it,
Alright, we'll leave.
but we, yeah.
And this is going to jar 1. Stand by. Stand by. Index. Two. 1. Okay. So we need a better picture for
Yeah, just any of the sediment on top here?
Let's look at this picture.
Just
So do you want just the surface layer or
Just
dig
the
in
no,
as deep as you can?
just the surface.
The surface
Just
layer.
There's probably not much
Bueno,
about
ahora podemos observar que con el succionador, llamado suction sampler, se va a mostrar una porción de sedimento sobre la roca que es muy interesante porque puede contener bastantes organismos. Por tanto, esperamos
Yes,
que
can we just like record that?
Yeah.
Okay.
esperamos que haya diversidad en esa zona que se va a aspirar.
Vacuum it.
Yeah. Okay.
Y esto va directamente a un contenedor especializado para ello.
I just got it on slight
Ahí vemos que va a
section.
funcionar la aspiradora de sedimento en este caso.
And dig in a centimeter. Are you happy with this one? Not too deep. You want to go a little bit deeper? It's rock and Patricia.
Yeah, it would be good to go a little bit deeper, but I think it's rock. It's 100 feet in rock. It's just not going deeper because there's no possibility.
Yeah. You can get as much as you want. Maybe I'll come down to the top.
Yeah, I think so. Come down.
In the corner, there are more
Yeah, it
sediment.
looks there's like a little bit of accumulation here, perhaps. Or here. I'm not sure, but looks like it.
Yeah, no full suction. Full suction. No, it's very thin layer.
Yeah. Maybe over
Maybe at the base.
corner.
Pull up. Got our bungee just bungee line here. Spin this around a little bit. It's gonna help.
Let me give you a better picture and sit.
All right.
You wanna just dig in,
Yeah, I'm in there. There we go.
Bueno, justo estás mirando en ese rincón que se forma entre las dos rocas para captar más sedimento.
It doesn't seem that we are getting much. Oh, we are getting some, yeah.
Number one.
You can see on the camera. Cleaning the house.
Just like
Totally.
Sundays right now.
Yeah, exactly. Feels like a Sunday now.
Una fiura que se observa ahí, probablemente una fiura de 6 brazos. En algunas de las especies solamente tiene 6
Yeah.
brazos.
Wants to start.
Can come with us.
También se fue la fiura hacia adentro.
It's very thin layer of that.
Yeah, it's what it is.
Probablemente la especie Ophiacantha vivípara o similar.
Maybe at the corner. Maybe that one. Corner of the walls.
O Ophiacantha Antártica, porque hay varias especies de 6 brazos.
I think that's it. There's not much there. Yeah. Now the last thing we need is a rock. So let's see if we can find a rock that we can grab over here.
Rock.
See him?
Up here.
Is that rock? Oh, that's a bubble.
I had one this much, but then I dropped it. Fell into the precipice there.
That's a bubble, right?
No, it's a bubble. Yeah, no, that's not a rock. No, but we can keep going and find a rock around. Just zoom out. It doesn't need to be in this particular spot. should be.
Is that a rock on the right? I think. Is that loose?
You can try.
Try. Where the fish is? Okay, I might have to move.
Yeah.
Let's see.
But I'm thinking
I don't know if we can reach that.
this wall is not gonna disappear, so maybe we find one a little bit.
Higher?
Yeah,
Okay.
perhaps.
Okay. We shall move on. Whoever goes ahead and lines. Okay. I'm departing here.
Bueno, podemos ver que el vehículo está saliendo de la zona del... donde estaba colectando. Para seguir con nuestro camino.
I'm gonna swim a little distance away from it for now, Patricia. Just to let
Okay.
you know until I'm sure I'm gonna open it. So
Yeah.
it's
In any case, yeah. As soon as we find a rock, we need to take it.
first rock possible.
Yeah.
I can go near. You can see the top now. Could break one. Or go higher.
Well,
Rosela.
the first possibility that we have.
Ah,
You see one that you're interested in?
I don't really, I couldn't tell. It's just as soon as we find a rock.
Keep an eye out for one. No. Find one?
Serious.
Hard to tell, it's covered in silt, but you know, there's some funny one up, it's just a little bit further down. That. I suppose that might be loose, or is that just?
Well, there's several too. I think they can fly off.
Yeah. This looks like
Yeah.
doable.
Turn a little bit. Finish your hand. Come on. Okay, let's just see it stable. Alright. Let's see. All stable. To get you a better picture. There you go. Zoom in. Got it. Sure safe.
Y se puede observar que el brazo está intentando sacar un trozo de roca para muestreo también.
That's not helping at all.
I think that will break.
On this. Oh.
Se ve todo muy sedimentario.
Well, they're quite crumbly.
Is there anything else? Maybe from the bottom?
It's interesting. It's interesting.
I wouldn't grab it. Just for fish. Just try to. Do you want this picture? Sure.
Yeah, put it in the rock basket. It could be any of these.
Or it's kind of like, I don't know if it's now or you just want to get down.
Ah, yeah, that was me.
This one.
Mm-hmm.
So if you try and put that one in, is that okay? Yeah, why don't you just drop it in there? Yeah. Alright, now which one are you talking about first? Let's think of it. Any of those? Maybe this one? Different nose? That one? Just close your jaws and then just kind of see if it moves. If it moves, then you open the jaw. Okay. Just kind of poke it. The other side, take it out. Yeah. Stand by. Stand. Okay. We're gonna call this one name I said.
No te pillo el piso.
So, characterise science scan. Not moving. This little guy. Doesn't moving. Keep that down. What about that? Go on the side. Go on the side, go on. Oh, they're all moving. Oh, now you got it.
Ay,
Wait.
Como pueden ver, hubo una reacción unánime de
There's more.
Well.
una
Let's try the next
reacción
one.
de asombro cuando cayó la roca hacia abajo, y como es una pared no sabemos hasta dónde pudo llegar.
They're very crumbly.
Oh, there. Oh, no, one more. No, stop. No, keep going. Wait. To that beast.
Well, let's try one more time. If it doesn't go, we'll find another one later.
Big one. Yeah. That one. Go on over. Flip that.
Tercer intento.
I think they are too crumbly. It's not really going to work. So I think we can just continue until we find something more feasible. I think we probably got a good piece in the basket.
Okay. Ready? Yes. Okay. That is shoga. Ready when you are. Take me off. Okay. Nice. Almost over. How about that one, Patricia? Is that what those two?
Not really.
right? Yeah.
It will crumble anyways.
Okay. We'll keep going on. Climbing up. There's a lot of rocks on this side.
Can we zoom into? Oh, let's let a little bit up and then zoom into those corals here, so that our colleagues can have a better look. But let's go a little bit up, so that we actually let's go a little bit up, so that we have a look of what is there. Have a good overview.
Esto van a hacer zoom en unos ejemplares de coral al lado de una asidia gigante y sobre la asidia otra ofiura.
Go ahead and operate the camera. I'm going to just do this on the fly. Yeah. Unstable.
Okay, let's get a nice close up and then we'll get a piece. Yeah, it comes with a little body if you're lucky.
Como pueden observar, se ven los pólipos claramente. Cuando hace zoom y enfoque, se pueden observar claramente los pólipos.
Happy?
Yeah, yeah.
Okay, shall we collect or?
We should.
You should collect?
Yes, please.
Thank you. So, which ones are we going to collect here on
Gonna collect this one.
Roger.
Yeah, about 10-15 centimeters. If it's a little bit more, it's fine.
So, we're going to go in coral river.
Yes.
Favor the light. Got it. Works for me. Stand by.
So we're gonna collect this one.
Okay, what else are we gonna collect on the left?
One on the left, but it's gonna be a zoom in. We are not sure if we actually want it.
Yeah, I'm just gonna position a little bit more closer to the left.
Perfect.
So I don't have to again this hole. I guess it's that small coral thingy on the left. Well,
Yeah, we'll zoom in.
go ahead and zoom in, T.
Zoom in here.
Kind of stable.
It's quite similar to one we collected the other day. Have a look. It's quite similar to one that we collected the other day. So let's collect the other one. Yeah, that's fine. We're gonna collect this one. The big one. Big one, 15 centimeters. So.
Got the camera?
Pull the lid off number 1, I think.
Yeah.
No,
transect. Maybe we put one in something before and something after that.
No, number 2 has already one. It will be great if we get a branch that has this little guy. So there's someone living here.
Okay, he wants a little number. So
I want a bonus, but if it's posted, don't worry about it.
Can we get it here somewhere where it's a little bit
Fantastic.
thicker? You got this thing? Because we want the
Veo
base.
que están intentando cortar un pequeño trozo que contenga también un peracárido que está sobre la esquina, sobre el extremo.
Cannot be.
Yeah. It's enough, you think? Yeah. There's an animal. Is that an animal?
Yeah,
you know?
exactly.
Okay. So once you grab this tea, there's an animal, so be very gentle so it doesn't fly off.
I mean, if it does, it's not a big deal, but it will be great to have the bonus.
Yes. Okay. I don't think this is gonna fit in the crib or hold on.
No, it's too much.
So we'll put it in my box.
Also, we don't know. We can take a smaller piece. It's fine, even if it's hard to get the animal.
Okay. Try to separate this branch out. Okay. There you go. That's good separation.
Oh, that's wonderful.
It's a master.
You guys are great.
Oh.
I take back what I said.
Never mind. But we still have this one. It's great. But as I said, if we don't get it, it's not a big deal. The priority here now is the coral.
It's on the plate. Yeah. Just needs to go crawl and jump. There you go. It's on.
Oh, okay. Totally one also run away.
se acaba de desprender, parece.
Do you think it will fit in the firebox? And in the closet?
Yeah.
Gonna quiver.
In the quiver. Great.
That's about as much as I can help with. So. Let me see if I could maybe have a bit more light.
Coral one.
There you go. Very good. Well, that's good. Nope, you're not in yet. So you have some picture now. There you go.
Looks like we got something on there, right?
Yeah.
Roll in there.
Okay, you roll too much. Let's see. There you go.
See if I can be able to drop this down.
There you go. There you go. Oh yeah, watch it. Careful. You got most of it.
That
This
one in, right?
this one- Yeah, so most of it went in, now you invert this one.
Can I have a request? Sorry.
Stand by. Let's let him finish this one.
Do you, sorry, one question. Do you have a good grab of it?
Yeah.
Because I forgot a clamor shot of the,
Oh,
of the
sure.
thing.
Oh, yeah.
That was, okay, that's good. There you go. Perfect.
Great, thank you so much.
Is it in focus?
Se pueden ver perfectamente
It
los
is.
pólipos.
Yeah. Great, fantastic, thank you.
Okay.
Ciao.
Okay, and it came with a bonus polycube.
So, you're gonna grab another coral, right?
So again?
You're gonna grab something smaller?
Yeah, when I take this starfish.
A starfish, okay. You wanna put that in a cooker or?
Let me give me a second.
Yes.
Arriba?
Yes.
The one on the top?
One on top.
Can be in a firebox.
Or okay.
No, it can be in a firebox.
Okay.
making sure nobody escapes.
Okay.
So it'll be,
You
yeah,
want
this
that
one
one?
here.
Yeah.
Wait, yeah, that one.
Just want to grab out. Like,
Yeah, I'm just gonna peel it off too. Peel off on the leg.
Master T. Okay, which white box do you want this in?
4C.
4C.
Actually, it can be 4A even.
4A. Watching out.
No, that is 4B, sorry. It's a sponge that can be fragile. So it will be 4C.
Okay.
4C.
4C final? Or 4C is the
It's
final?
not there yet.
C, B,
No lo apunta hasta aquí. Yeah, 4C.
C. Okay. That's the picture. Along the way. Chip up. Right there.
Yeah,
Beautiful.
4C.
4C. Okay, the next one, Patricia.
Next one is keep going.
Keep going.
Alright, that is Fort Marshall.
Okay, here we go.
Go, go, go.
Now we look for a rock.
Alright, the rock. Most elusive, hard to get thing on the sea. Quite
Hard
bad.
to find a rock.
Yeah. It's kind of hard because they're very crumbly here.
Yeah, they think it's loose, seems to go all the way to the bottom.
Okay, here we go.
Maybe near the top is easier. I'm gonna zoom into this guy, just a zoom in.
Roger.
Maybe this one better,
Okay.
more accessible.
T?
Yep.
See that pink thing?
Yeah.
Go ahead and zoom in. Wide. Let's see.
What's that?
Coral.
It's a coral.
Yes.
And I'm just wondering if we should collect it. We're gonna collect a little bit of this one.
Okay. Coral quiver. Okay. In position.
Bridge, ROV.
Ruby Bridge.
We're gonna do some more collecting, exploring here on this wall, just checking and see what our iceberg situation is.
Yeah, no worries. The longer we can be here, the better. Iceberg is off the starboard bow, a few hundred meters, and moving to the northeast. So we wouldn't be able to go to the next 3 waypoints if we were to leave now.
Que se tiene que mover nuevamente.
That is good update. Cool. We'll pick some stuff up here on the seafloor.
Excellent. Yeah, it's moving quick. Maybe 0.3 knots. So hopefully we can just hang out here and then it will get out of our way.
Roger that, thanks.
Thanks.
Okay, we have a nice playground here, so we will not get bored.
We can as well. Give him
So let's get first a good close-up of the polyps.
Cam stable.
zoom.
Yep. It's
And also, we want to get some glamour shots for Alex too, if we need to use some time. Some glamour shot. Good. Collect everything. Collect it.
when kill the lowers.
There.
There you go. Slide there.
Magnífica vista de los pólipos con fibroides entre medio.
Decrease the ISO a little bit.
So that's
That better? Color ones.
that. This is good for us. If this is good for us.
Okay, he's good. He's happy.
Really good.
He's happy. I'm sorry.
I'm always very happy.
Yeah. So, do you want to collect like a piece at the
piece.
bottom or on top? Maybe
I
at the
mean, in
bottom
this
might be
case, the
easier.
bottom.
Yeah.
Some of these pieces are in this branch.
You want to open a quiver, right?
Yes.
Five, you said?
Oh, I said live. About 3.
Three. I'll give you some light. Lower speed. Okay. Point. Okay, I'm going to zoom out so you can position on the way you want it. And he said bottom right. Yeah, that's the bottom. Can you get any more horizontal?
Yeah.
Yeah, their jaw a little bit more horizontal so when you cut it straight. If you can.
How much did you want pictures? About 10 centimeters down?
15. A little bit more than that. Just a bit more than that.
There you go. That's awesome. More inward. This is there you go. More inward, and then that's just give you a better picture.
And this will tear too, right? So, really want to do it.
Why are you cutting like that?
It'll just rip, right?
You know, if you cut it like that the piece, the bottom piece is gonna fall off, isn't it?
I think the stock is the birch needles, right?
No, the base of that one is above you. It's here. Can you mark this? It's here. The base. So if you cut
okay.
this here, this thing's gonna fall off.
Or are those two different ones?
Yeah, you're right. I didn't notice. So, well, Well, maybe we can just pick this one.
Yeah.
If it's visible.
I think you had it
Yeah,
right
that's
the first
cool.
time. The base is actually here, right? No, the base is on top. I would invert that blade.
But I don't need the blade to cut it, right?
Yes.
You need the blade to cut this one.
Yes, cut the top part.
For us, the ritmo is about this.
Yeah, of course.
Oh, I'm sorry. I think we were talking about two different things. She's thinking of cutting this part.
Yeah, that's what I was thinking.
Bye.
You see that he's having a bad day.
This part. Yeah. That's what I was going for. What were you going for?
I thought you were trying to get the other one. That's why you wanted the blade up.
Yeah, because I want to pinch it off like this.
Okay, try it. Do the shots. Watch, I'm getting pushed.
This one comes with an amphibot, but I don't want to say it because as soon as I say it, it falls off. Oh, and an isobot, but too many bonus.
Too many bonus.
Sorry.
Yes.
You gotta cut the wrong way. Invert your wrist.
Yeah, I'm not trying to cut it, I'm just trying to pinch it off.
I'm just trying to pinch it off. I don't know if it's gonna pinch me.
Let's do... Alright, let's try to cut it, but...
Zoom out a bit.
Atrás se observa una magnífica ejemplar de Ophiocantha vivípara, probablemente la especie que incuba, de la especie de Ophiro que incuba.
You got sit picture too. Yep.
Glamour shot, please. If it's possible.
Before everything swims away.
Wonderful.
Okay.
Well, it feels it's gonna fall off, so it's almost the priority is to get it.
Okay.
So it's just
Hold your photo for a second.
Yeah.
Vemos que trae adosado también al coral, trae un ejemplar
No.
de estrella canasto o estrella organocefálido.
The isobot is about to. The amphibot is about to. No, it's wonderful.
Probablemente
Yeah,
de la especie astrotoma o similar. Y un isópodo. Oh. Va directamente al depósito de corales. Esperemos que no se desprendan los ejemplares. Number 3.
It comes with so many friends.
Y muchos ejemplares de Ophiora también trae.
Uy. Uy. Oh. Well, who could blame him? Oh, well, my friends are gone.
Lamentablemente vemos que se escapó el isópodo justo en el momento preciso.
But the most important thing is we have it
The corals.
safe. Now that's safe in the coral quiver. Thank you so much. It was a nice one.
Alright, we'll get you next up on one of these flashes.
Happy. Let me have a look. Let's have a good look of what is around. Not for the moment. I see anything over there. Can we keep going up the wall?
Again. Alright, here we go. If there's time, would you mind if Alex and Brooke came in and tried to get some shots?
It's fine.
Okay.
Can we zoom in to the corals? So this one first, and then that one, and then that one. Can we zoom in this thing first? Well spotted, cap coral. Thank you. And now we're going to zoom in the other corals. They were yellow. This one. Going up to the side where we want it. Let's zoom out. And zoom into the other one, which is, so we'll zoom in this one and then in that one. And then in that one. I mean, I'm not sure if we want on the top is the coral, but
Salida Ozoan.
it's a beautiful one actually.
The pink one or the white one?
Pink one.
Pink one.
I'm not sure if it's the same. Let me spotted before, but let's give it a shot. And then let's move on to the next one. This one here. Do we want any of this? No? We already collected these ones, all the three of them, so we are gonna try to have a look. I'm not sure if the other one is coral, but I meant, no, I don't see any coral. Maybe that, no. Whatever it was not a coral. I cannot see it anymore. It was probably just the hydrozone.
Hydrozone.
And I guess Alex wanted his shot before we leave this rock. So
He said he's on his way.
he's here.
Oh, he's right there.
Okay, thank you very much.
Yeah.
Always a pleasure.
Thank you.
See you soon.
See you tomorrow.
See you tomorrow. Okay.
Water's murky, as you can see.
Zoom into that one. Here. Here.
That brown wall.
Yeah, let's zoom into it. And so we want this one? It's the same one.
Which one, the white one?
Can we have a close-up of this one?
To
Just
your
a
right
close-up.
there. No, further right.
No, but first we want to have a close-up of the fish and then the coral. Just to see it, have a nice view of it. It's a beautiful one.
Perfect.
And first,
Thank you.
Five minutes to film. Yeah. Okay, cool.
There's a huge iceberg just rushing up against the bow, so.
Nice. We have a check from the food.
Yeah, no, that's quite but apart from
So still we want to
make some progress. Yeah.
So I'll make it close up. Just to fly the camera,
Yeah.
take it out. That's just fine. Some drop on you.
There's something.
Do you want to go down? That one. That one. Okay. Okay, just can you give a stick and a rubber stick?
Yeah.
Just point me where you want it to go.
Alright. Embrace your never up there. Alright. Stop right around.
We're gonna swap pilots. Can stay for 5 more minutes?
5 more minutes, Roger.
So back up, set, and that is the target, primary target.
Yes. We got about 5 minutes. That's fine.
Okay.
Camera.
Yeah. Now, rocket. I'm gonna keep that in the center. Moving in. Easy, easy. Very dramatic. How are we doing, Alex? Alex, how are you doing? Okay.
Early bridge.
Here comes John. This is early.
Hey Tyler, I'm gonna have to start moving. The iceberg is getting a little close for comfort, so I'm gonna have to come to port and probably ahead a little bit if that works for you guys.
That works. Is that going to be an immediate move then, Luke?
Yes.
Okay, we will immediately start moving. Good.
Great, thank you.
Flying on the wall.
Sorry, I'll shoot you the text. What's up?
Sí.
So the iceberg is coming. Yeah. Yeah. Got it.
Sí, 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 lo vi, vieron una foto ahí.
Yeah. And then if you guys get any more close-ups of cool stuff, probably that's cool for hours. He's got whenever you're done moving. Thanks, fellas.
So what's up with the, what's up with the temperature?
What's not?
Temperature's not working anymore?
Sure.
Honestly, you're on a shit face, so there's nothing.
No, I do appreciate it. Oh, there you are. Where's that camera?
It'll be off the starboard side now if you want to show the iceberg.
It's not what it is, 20 meters. 30, whatever.
For you, so you can see how close you are to him, to the red circle. I'd get right on it. Yeah, looks like he's going hard for southwest.
Oh, it's going fast.
Yeah, he's going fast, man.
That was a kidding.
No. What's that?
Oh. We should go under.
Yeah.
Just to see how the ice.
Just outside the red. Inside the reds. Slow it down.
Yeah, hi everyone. We're currently diving in front of the Bach ice sheet in the Ronne Entrance in the south of the Bellinghausen Sea. At the moment we're dodging around an iceberg, which is why we're not very close to the seafloor and filming. Once we're clear of the iceberg, we'll return to sampling. Buenas tardes a todos. Estamos aquí en una inmersión frente el hielo, la plataforma de hielo se llama Bach en la Ronne Entrance en el sur de Mar Bellinghausen. En este momento estamos tratando de evitar, hay un tremendo iceberg cerca del buque, entonces estamos haciendo la maniobra para evitar que chocamos con eso. Entonces volvemos a mostrar en un momento.
Over the bridge.
You only give me one thing at a time. Go for RV.
Yeah, we've probably got enough distance here. Are you guys interested in sampling where we are or going to a different location?
Anna, do you want to look at those photos?
Stand by one.
Stand by.
Yeah,
You just can't go to starboard any. The large icebergs right abeam of us.
Yeah, we see it on the
Yep.
Can we have a look at the pink?
No,
Yeah.
the white one.
The white? Which one? This one? Yeah, this coral here is the one we're interested in. Let me have a zoom in first.
Yes, please. Otherwise, it's not go the wrong
Yeah, yeah. Stability.
Yeah. Yeah.
Okay.
Want to sample it?
Yes.
Yeah.
So this is the maximum.
Is that maximum zoom?
From here, yeah.
Yeah.
Okay.
Yeah. Sample it.
Yeah, okay. So if we coal quiver.
Did you see?
Okay.
We need to move closer.
I mean, the wrong.
This far. We need to keep changing all around.
Reckon if they run in, we'll be able to, or it's not.
I think you're gonna have to move.
Okay. Okay. Which blue is?
Huh? Which school? There's 5, 6, 7 or 8. It's for the back full. For whichever one's easiest. No, 5 is fine.
You want the lights on?
Ah, yeah.
Can you connect this one? Can you zoom in on the other one behind to see if it's the same?
That one?
No. This one. Okay.
Yeah, okay.
Yeah, please.
The one on the left, you want to look at that?
No, collect this one first. Yeah.
Yeah, they want to collect.
This one here. This one. One thing at a time. Okay, can we have a glamour shot, please?
That's all this one.
Ooh, amphipods. Yeah, thanks. Yeah, we close that one. We get the other one. Yes. That was number 5. Yeah. So the other one was that one there. We have a zoom in, see if it's different.
On the back side?
Yeah. That. Is that different or the same? It's the same?
Yeah, but I need to read.
Oh, okay, well, because we're going to have to move to get that one, aren't we?
Is it okay?
Yeah. Yeah, that's fine. Yeah. We want it, so.
Alright.
Yeah.
Do you want that same crawl through?
No, in a different one.
Yeah.
Come around, I guess. So yeah, number 6.
Sorry.
Number 6, I think.
Yep.
Don't think it's necessary. It's the same species as the last one. Anything else before we move on? Oh, the cup. Is that a cup coral?
Different color or same color?
No, we're not collecting it, just want to zoom a picture of it.
That's the dive number. Is that correct?
Yes.
Yeah, that's the dive number. Yeah. Just to make sure that it's the right one. 718? Okay, good.
Yeah?
Yeah, perfect, thank you.
Right. Nothing else?
If there is, I don't know if we are on time or not.
We're never on time.
It's because I need 3 from the same species.
Yeah, but that's definitely different.
I know.
Yeah.
It was just because you're zooming in and I'm not sure if there is a
That one.
there.
What, that?
Yeah.
No, that. Which one?
Here.
Well, we'll have to move around the other side because that was the limit of the zoo, I reckon.
It's just I need one more.
Can we swing round to the other side then with the rod? Because it'll be one of these.
Yeah, we'll do it.
Yeah, I think it's this one.
No, but it's an idol.
Yeah, I know. We're zooming behind. Like, so this. There's no possibility. We don't. I just
Yeah,
need
so
one more.
if you can move around that area. What? There was one just been like that one about.
That one?
No, we see it.
Yeah.
I see it. It's not on there. Yeah. But there's something under the rock, so. Yeah. Yeah, well, as I said, there was more on the other side, so I can't see here. Ah, yeah, rising up behind. Yeah, one right in the middle.
Oh, I think that will get some. You have to put that thing just to the left of that one, push it and slow, maybe not come down a lot. Push it. Like this? Yeah, come back, down, take that. Put it right there. Alright. So it's supposed to stand out, push it in, we're not gonna be touching it.
For this one, right?
Yeah. Go.
Yeah, so quiver 7, I guess. This game?
Yes. If you have
Yeah. Right. I guess, can we ask the bridge if we can move northeast back towards the track now? Or?
So we're gonna have to go
North and
west.
then
We're gonna have to go west.
Yeah.
And then basically go around, because right now that iceberg is right off starboard side, right off midships, and corner of the door.
Okay. Yeah. Head northish.
So,
Yeah.
is that what you want to get back on? With the
Eventually we
track?
want to head back into the track originally once we're around the iceberg.
Go around west. Bridge, Harvey.
Early Bridge.
Hello. Yeah, can we maneuver around this iceberg, like I guess go up to the west and then to the north and work our way back to the track?
Yeah, we can do that. I can start coming ahead and to starboard, and then yeah, we can start to head back a little bit east if you'd like.
Yeah, once we get clear of the iceberg, we'll come back to the east and put the waypoint up for reference if you want.
Yeah, sounds good. Do you want me to just start coming slowly to starboard and a little bit ahead to clear it?
Yeah, you maneuver how you have to, and we're just gonna follow you.
Okay, perfect.
Yeah. The lasers on.
One sec. Yes, they are.
Yeah. And have we lost the temperature sensor?
We don't have one.
There's usually temperature on the
We haven't had one. It's dead.
Okay.
Hasn't been on the whole cruise.
Oh, cruise.
The high temp? Yeah.
So what's the temperature in the seawater?
That's from the CT.
They had to turn the
Which
CT off
had to
earlier.
get turned off because of gravel.
Oh.
So no, yeah,
So
no more this
right down there. Nada. That was gold.
No, I guess not. Huh?
Early Bridge.
Go for ROV.
which waypoint are you guys wanting to go to next?
So I've got the Falkor 2 waypoint up there. If that's not far enough to the north, I'll move it up the track line.
Okay, I will aim for that. I'll have to come a little bit ahead first, but I'll aim for that. Thanks.
Other copies.
Might be cold outside, but not too hot.
So put on the oven right now.
¿Qué pasó?
Don't give it Yeah, it doesn't. Now you try to, once you're here, it's not like, a show or you don't give it again. So basically put it where you want it, and then touch it. And I found that actually, if you have that with the
Okay, yeah.
Yeah. Just gonna follow him around, dodge around that thing. Work our way back. Just looking at it, we'll probably miss 5. Have to go
Yeah,
6.
no, it's fine. Back to it around waypoint 6. One bit of seabed is as good as another.
If you see a big white wall come to play.
That's the first decent bit of rock we've had. It's groups.
Isn't that the goal, isn't it, to follow that ridge down?
Yeah, basically. I guess unless we see something special, we'll just keep, video transecting for a bit. Yeah.
Stop. On. There, just there. I was just thinking about those when we showed up.
Yo, nice.
I think we will slurp that.
You want to collect that?
Yes, please.
Bridge, can we pause the move, please?
I'll stop.
A slurper, yes, I think. We'll stick it in number 4.
Same species as the one you always sampled? Is this the same species that you always sampled?
Yeah. What's the other one?
Wonderful. June. Okay. That's number 1.
Now cycle it. Keep cycling up. No, touch one. So when you're doing it, when you hold it down, it'll actually come back around next to where it needs to be. And then you let go and let go. We can try to stop it midway to where your heart is. So we'll keep trying to go back and forth. So it gets all the way. It's good flow. This is what you want to know.
Yeah. It's a biggie. Suck.
Don't do the noise.
There he is. Pretty sure that's in that here.
There he is.
Yeah. Oh, excellent.
Which
Yep.
sample?
4.
Four.
bridge, are we?
The bridge.
Yeah, we can continue that maneuver around the iceberg, please.
Copy.
So you want to,
So that waypoint is just a reference for him. He's navigating around that.
yeah,
So you may not get to there. I don't know.
I guess once that iceberg disappears from view, then we can start heading back.
Right, yeah, once that thing gets to the stern of us, then we'll actually be able to start doing it.
Yeah. Well, anyway, the track comes back across us anyway, so we're fine. There's another one, but yeah, we've got one. Just put that as an observation, not too much. One is okay. Yeah, all that. Destroyed population. Chanchito. How steep is this slope we're going? You can't tell. Ben seems to think he can tell. DJ that master or something. Yeah, it's, I think, good one's vertical.
35
It's just because we're obviously very close to the
it's a proper wall.
altitude is over 4
So you
meters.
can look at that.
Yeah.
So you can see it's going like that. We've got a 6-degree down. So.
Yeah. Well, I think it's just gone very steep, hasn't it? That's down.
ROV bridge.
Go for ROV.
Yeah, just to let you know, it could be a very long time until we have access to waypoint 5, 6, or 7. The tides are starting to change as well, so it could change the direction of the iceberg movement.
We can just go north.
Yeah, I guess we'll
We'll just go north. We'll skip to like say 7.
Yeah, that should be doable.
Bridge ROV, that's updated waypoint.
Great, thanks.
Vad mycket tråkigt så här. Åh, vi tråkar på honom. Det är ju tråkigt. Säkert du råkar.
Mm.
Yeah, because I can get back on track, I guess, because we're going off in the opposite direction to the
Yeah. Ajá.
Después la términa vive enterradita, entonces puede venir un oso bollito.
Ya, bueno, cuando está como los saquitos.
La términa.
Sí, sí.
Pero no, obviamente sí, tú.
No, es un poco de enterrar.
¿Quieres que diga algo?
¿Ah?
¿Quieres que diga algo?
No, está hablando sobre roca, pero un poco grande.
Yeah, they're gonna stop. Just come back and stop pretty soon.
Yeah.
Just let them get a little bit closer to us. Zoom down there.
Yes, from here west is upside. Well, yeah, the tracks. So I guess we head up slope from here. Don't we? More rock. Only a little bit. Anything loose there? What about that one? Yep, cool. Great. Is it a rock? Is that a rock? Es un rock. No, pero ese es sólido. Sí. Pero ese es roca. Yo creo. Sí, sí. Yeah. Can we pick up that rock?
Inboard.
Outboard.
That'll be it. Great. Is that nice? That's just not a rock.
It's not a rock? Not a rock. Oh well. Bullet mission. Can you just try see if the one next to it is loose? Or
Which?
The one just to the side here. I don't know if it's part of the bedrock or loose. No, I think that's part of a much bigger rock. Yeah, okay.
Yeah. What do you need?
While we got it out, can we get a back sample? Number 2 should be free and still in the basket. No, try and fill it back if you can.
Oh, you know, just on the top of the pit?
Well, the surface, yeah, but I think the drains is short sweeps. This doesn't appear to be bulldozing quite as much. Famous last words. Everything we're interested in comes up from the centimeters and the sediments.
Just go right there, jump.
Looks like quite a bit. Yeah.
It's too much of a slope.
Yeah, that looks like it's about half full. Well, yeah.
I can speak from this other side.
Okay. At least the current seems to be blowing most of the sediment away. Won't move, will it?
She's not moving.
It's in my territory. I ain't moving for no one. Yeah, that's looking good. Yeah, I think so. Yeah, thank you. Yeah. Okay. Yeah.
So
get to one.
Bag number 2.
That was bag number 2. Yeah. Yeah, and we're going to do such Yeah, so we suction up some of these brachiopods that are on the rock. This should go in number 3. I'm on the other look. Yeah, okay. Yeah, we're at number 3. Yeah, any of these around.
Yep.
Yeah.
All right. Sorry.
You might have to nudge him off the rock, but esto piensa que está suelta, puede ser. Ya, ya. ¿Cuándo terminamos eso? Ajá. Esa es blanca. Ah, con la scoop. Ya, ok. Ya, yo. Mira una. Un picnagonio. Really? No? Ah, yeah, okay. Si queremos hacer nuestra. Yeah. I think that's enough. Okay. And we want to see if we can get a rock from all that mess. One of them solid enough to pick up. I think everything's through. Not sure about that one, but we'll see. That might just be sediment. Yeah, no. Oh no, looks solid, at least at this end. Yeah, well, this is a rocket. Yeah. It's fine. See? What about that one? Is that one loose enough? The rock's got a mind of its own. No, it's really, yeah, it's really soft, isn't it? Ooh, what's that? What is that? It's like sandstone, isn't it? No, I'll stick it in the basket. You've got it now. Yeah. Looks sandstone-ish, doesn't it? From the days when Antarctica was a desert.
What do you want this?
Just stick it in the basket.
The basket, all right.
Is it the same divestream or something?
No. It's not really free. Seems more solid, this one. It's kind of oxidized. It's like oxidized quartz or something. I don't know. Yeah, thanks. Let's put that in the basket.
That's a nice sample, thank you.
Alright. Let's head towards where we're going to be. Geological something?
bridge, ROV. Updated waypoint, please.
Updated waypoint. Updated waypoint.
Thank you.
Hold on, is that the fish that Tom wants? That fish.
That looks like, to be honest.
I'm no expert, but that looks like
But
the
I
one.
think
he
that's
sent.
what he said to me.
Sleeping fish. Tom says yes, please.
Okay. You want to fall back a bit and then come out to starboard, look at the net. And we'll try to net it rather
Okay.
than suction. I think he'll just swim away.
Okay.
Right, so let's come out to starboard and I'll grab the net. And then we'll try and net him.
Let's come back down. Oh. No, he's gone off. Keep it in hand for a while to see if we can catch up with him. Usually don't fly up. Go too far. No, I don't think that's him.
There he is, right there, to the left.
No, there wasn't.
To the left.
It's Darius there.
Oh, okay. That was following the ground one. You already know, so we have to.
Oh! He's not dumb. That's
He's not going down Sakito.
Where is he?
Tom says thanks for giving it a go. There'll be other opportunities, I'm sure.
Alright, so should we just continue moving to the
Yeah, kind of towards 8. At least we got video of you swimming. So.
Alright, moving on.
Okay. Yes.
See what else is there.
Oh, have you still got the net in your hand? There's another one, I think.
Yeah. Ready? Before you go in, no. I need to get this stuff out of the net. So
Oh, we've got some sediment. Shows how sticky it is that it gets held in by the net. Something in the corner. Looks like an anemone or something. Good eyes.
Das ist is that biology or something? Or
he's been buried alive.
No,
that's
that's
it. I guess we stick him in Biobox
Biobox
2.
or
2A.
Okay.
Don't Biobox? Biobox?
I'm out.
Forward to.
Yeah. Oh. Alright, frozen. How's that?
That's it. Number 2, right?
Yes. Either side, they're both empty.
So,
Yeah.
huh? It's a lot of water or is
I hope so. you can leave alternate?
Yeah, no,
Okay.
yeah.
What do you want to do? You want to check the map? What do you think? What else happened?
No, como site.
I didn't see enough to say. I guess we stick it in hope.
Can we do it? That's the side here. See if that fish actually swam out of the net.
That's it, Sit down, yeah. This one. What do you want to do now?
Just stick it in hope that it's in there.
You want me to wash the mud out, or?
I think
Alright.
no, we'll just cross off. But it was some other fish. That's what I asked. it doesn't really close with a net in there, but that's fine.
That's as far as it's gonna go.
Yeah, no, I think it did that last time. We just can't put anything by escape in there.
Alright. Yeah, watch anything.
Morning. Not quite. Thank you very much.
West.
Maybe we got a fish.
Maybe we did.